Looks like it's loaded, so I can add it.
And we are live. We are live. This is exciting stuff, Bob. Welcome
Nobel
everybody
Prize.
welcome everybody to the January 2026 edition of the Broken Science Initiative Journal Club. I've got Bob Kaplan with me. My name is Pete Shaw, and we're really excited for this edition. This is the first time we're going live on the Journal Club, and the rest of the team hates me for rushing us into the live format, but I couldn't wait to get this presentation in front of everybody in the extended world, not just the BSI world, because this month we're going to be reviewing one of Bob's papers. Bob's fresh off the press or hot off the press publications. So I couldn't be more excited about it. The title of the paper we're covering here is called Hereditary Cancer Syndromes Linked to Oxidative Phosphorylation Insufficiency. So if you're tuning in with us and that was a mouthful for you to understand, Bob is going to spend the next few hours going through all the details of what that means. But Bob wrote the paper. He's the primary author on it with Thomas Durig, Derek Lee, and Thomas Seyfried of the Seyfried lab, also in Boston. And if you're familiar with any of Seyfried's work, you could guess that this paper is touching on the metabolic or the mitochondrial metabolic theory of cancer. And what's really, really cool about this is that the paper is making a link between these hereditary cancers that are known and making the strong case that all of these genes that seem to predispose people to cancer have very strong metabolic links and affect the mitochondria directly. So am I right on that, Bob?
Yeah, that's the story pretty much. Yeah, we have, so actually, like a little bit of background. Tom Seyfried, I met him in like 2011, but I took his class. His class is called, surprise, surprise, Cancer is a Metabolic Disease. He teaches his class at BC. He also teaches like Biology 101. And his background is actually in genetics. I took his class, I think like in 2017 or so.
Mm-hmm.
And it was a lot of fun. Although there were a lot of students just thinking like, who the hell is this old man who's way too engaged in this? But one of the things that you do, or the big project or presentation at the end of the class, is all the students present. And Tom says, if you can convince me that cancer is a genetic disease, you get an A. And so it's interesting. He frames the class and he provides it's hard not to come away that there is a metabolic component to cancer after taking his class. But I remember asking him, I said, Tom, what do people point to? The people who we'll talk about the somatic mutation theory, but it's effectively, if the people that think that cancer is a genetic disease, what do they point to as the strongest evidence? And he said probably hereditary cancer syndromes, because those happen and those are literally defined by the gene mutations. We'll get into that. And they occur much earlier, generally speaking. So if lung cancer, if you look in the United States, median age for like a cancer diagnosis, I think is almost 70 years old. So half of all cancer diagnoses are over the age of 70. In the case of a lot of these hereditary cancer syndromes, it's much younger. And so it develops much quicker. And we see that we have these gene mutations. So I never did anything formally. Along the way, I just started collecting all of these hereditary cancer syndromes. I'd look into them. I just started creating like this gigantic document as I do, and collected, they say there's over 50 hereditary cancer syndromes, but I ended up looking at, there's something called penetrance, but it's just basically saying, if you get this gene mutation, what is the risk that you're going to get cancer in your lifetime? provided you don't get hit by a bus first. So like your normal course of your life. So we selected the most highly penetrant and also the most prevalent. So try to make it impactful. But because I probably looked at over 50 hereditary cancer syndromes, basically collected all those things. So hereditary breast and ovarian cancer, for example, and there's another one that's related to colon cancer. Those actually have, relatively speaking, a lot of cancer cases that are part of this hereditary cancer syndrome. Granted, it's still like 1 to 5% of all those cancers. So that's a little bit of the backdrop. We talked about this. I look at it like, can this convince me that cancer is a genetic disease?
Yeah, yeah. And reading the paper, like if anyone listening hasn't had a chance to read the paper, I highly recommend it. It's easy to follow. Bob lays out the argument in a great format. Each of these hereditary cancer syndromes, it'll drop down into the somatic mutation argument, and then immediately after, it'll go through the mitochondrial metabolic theory, and linking both of those viewpoints with the gene, with the gene mutation, right? And then you follow that up with a great discussion at the end, and lots of data, just looking at it from a 10,000 foot view. But if people aren't familiar with sort of the even deeper history behind this, like just before you get into the details, Bob, like in the early 1900s, Theodore Boveri was the one who came up with the somatic mutation theory. And then shortly after that, it was Otto Warburg that started to get people thinking about cancer in relation to the mitochondria, right? Warburg ended up getting a Nobel Prize, right?
Yes. Unrelated, though.
I think it's unrelated. Okay, yeah. So that was going to be my question. Is it unrelated? But he basically, Warburg showed that these cancer cells that he was studying, they undergo aerobic glycolysis. Right? And so basically, under aerobic conditions in the presence of oxygen, they're primarily driving ATP synthesis using glucose. And what Tom Seyfried's done is also he's added to that the glutamine plays a big role in providing energy as a different substrate from glucose in the cancer cells. Can you speak to that just a little bit before we get into the weeds here?
I could skip all the way to the end because I got like one of my pet projects. It wasn't in the paper, but I think people here will like it because I call it exercise and oncogenes. And so when you do high intensity exercise, I started looking into if they look at like transcripts, they look at genes and proteins. I started to look at like what lights up, because I think it's common knowledge that perhaps misunderstood in some ways, but if you do a fire breathing workout, you start producing a lot of lactate. That produces a lot of acid, and your muscles are burning. And what Warburg's talking about is that same thing. And he's saying, except in this case, there's plenty of oxygen available. Normally, when that would happen, you would see glucose go through the entire process. So you get, is it like 32 to 36 ATP? And instead, you're getting like 2 ATP. He said that's weird. It's seems completely inefficient, at least on the surface. And so he was the first to discover that, and he discovered a lot of things. I think he discovered NAD, so you have like NAD, NADH, cytochrome C. Effectively, a lot of the mitochondria he mapped out. And so the sexy field was like biochemistry for a long time. And then it was, I'm not going to remember the date, I think it was in the 1950s, that Watson and Crick discovered the structure of DNA. And everybody got into molecular biology. And if you don't believe me, there's an editorial by Watson just passed away, I think, recently. But he wrote an article in like 2011 in the New York Times. And he basically said, I'm concerned that I led people down. Effectively, I don't say the wrong alley, but in a way, he said everybody followed, and it was an amazing field. People still think it is molecular biology. And he started talking about metabolism, and he started talking about the Warburg effect. And I have books back here too, because he has textbooks like the biology of the cell. I figure it's like and he has volumes. And he mentioned this, that he said in his first volume, he didn't even mention Warburg. And this is probably in the 1960s. So he was well known and he had that work. There is like a mention in the second volume and subsequent volumes and things like that. But he did mention that. So anyway, you talked about glutamine. And so when we talk about lactic acid with exercise, that's a byproduct, or it's a byproduct of glucose oxidation. With glutamine, if you're burning glutamine, a lot of it in the mitochondria, glutamine goes through, and ultimately the exhaust, so to speak, is going to be succinate. And so if you see a lot of succinate accumulation, at least for me, that's curious. So I started looking at, and virtually nobody looks at this though. They all look at glucose and lactate. They look at fatty acid oxidation. They look at the crossover point, the crossover effect. And you read it in the article and it says protein is negligible. That's always the assumption, that we never burn protein because that would be stupid. We use it for, biomolecules and building things like that.
For those who don't know, glutamine is an amino acid. For those who don't know, yeah. So continue on protein.
Okay, let me see if I can find. Okay, exercise and oncogenes. Let me skip back that. Oh, that was it. Okay. All right. So this is I've got there's a few of these Nemkov papers, but you'll also see it's Inigo Samalon, and I think he works in some type 2 diabetic populations, but he also works in called performance athletes. And I don't know if he still is, but he was affiliated with, I forget which team it is. Pete, I don't know if you're a cycling wonk, but it might be the United Arab Emirates. It's the Tadej Pogacar. I hope I'm saying that right. I think he recently won the Tour. And so he was involved with that team, and he would study, I think he wanted to study these Tour de France athletes. And he's got a great line. He says something like, well, I think it probably makes some sense. He said, like, in order to understand pathology, we should try to understand perfection. And he thinks that he'll probably you're probably going to be hard pressed to find, more cool things going on, or high quality things going on, call it, with Tour de France cyclists if they start looking at their metabolism and start looking at their mitochondrial, or you want to call it mitochondrial flexibility or function. So he started going in there. And in this paper, they're looking at World Tour professional male cyclists. So I think they're like on the cusp of potentially like Tour de France, some of them. And so it's effectively like you're looking at like very high performance athletes. If you know anything about like how many watts you're doing per kilogram, you start looking at it, you're like, that doesn't even make sense. It reminds me of Kipchoge, if anybody like a Boston Marathon, I think of, that he runs a marathon in effectively 2 hours. And so that's crank up a treadmill, right, to 13 miles per hour.
Mm-hmm.
Good luck with that for 2 hours, right? I think they would travel around the marathons and they say, what's your Kipchoge number? And they have this, it's like a gigantic treadmill. And
people try to just see how long they last.
Yeah. And there's like a foam pit on the back. So if you can't do it, right, it's all safe instead of like in the lab, you're trying to like catch the people.
So these guys are using these high performers to understand metabolism. It makes sense.
Yeah. And so this is like the graphical abstract. They had like 4 different settings. One is a graded exercise test. That's fairly similar to if you've ever done like a VO2 max test, or if you know what that is. What they say is like a graded exercise test is you warm up, and then you have these segments, and they just keep adding load progressively, which is what you do with like a VO2 max test, to exhaustion. So until they can't do it anymore, typically. And then they have like a field test, which was like I think Inigo would probably call it zone 2. So that's his bag. That's what he likes calling it. So long, low, and slow, like all day pace aerobic, and that 5 hours, 20 minutes, which is probably par for the course for some of these guys. Then the lower left, they grab these biomarkers before and after. They race at different stages during a particular race. And then they look at like biomarkers, like what are the biomarkers that are lighting up? And I just wanted to see like what's going on. Is there anything kind of interesting to me? And let's see. So they have this one here, graded exercise test. They're doing lactate. So they're probably looking at like what's the lactate threshold, things like that. And so this is kind of like what they looked at with lactate. And this has probably been reproduced many, 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 many times. Mm-hmm. What's impressive is if you look at like the watts per kg, they're at between 4 and 5 watts. I think most people their lactate would be through the roof, and these guys have lower lactate. And that in part it's not just how much lactate you produce, but also how much lactate you can break down. And they're very good at that. I think Lance Armstrong is actually his VO2 max isn't like remarkably high or anything like that. I think, but he does boast that he has like an amazing efficiency to turn over lactate and keep that relatively low, which would be a huge advantage. Like when you start going, somebody's on the hill and they start taking off. So then you get the long aerobic training day. These are metabolomics from graded exercise tests. And at the top, you got lactate. And then here, this is the graded exercise test. And so they looked at like the TCA cycle, which is in the mitochondria. And when we talk about glutamine and where is that going, the glutamine can go into the Krebs cycle, or the TCA cycle. Mm-hmm. And it's glutamine to glutamate to alpha ketoglutarate. So you see the alpha ketoglutarate is still left of succinate. Alpha ketoglutarate to succinyl-CoA to succinate. And the thing is that these guys don't mention it. Like virtually nobody mentions this. I probably should have put a slide here, but it's textbook Wikipedia stuff. And when you look at succinyl-CoA to succinate, you make either a GTP or an ATP, and they're sort of equivalent, but you can make an ATP. And so you can make an ATP right there, not in the electron transport chain, not in glycolysis. And so there's this third pathway. I don't get into that fourth with you start talking about like creatine phosphate. But in this case, you've got this potential fuel. And this is, Tom talks about this, that the missing link with Warburg was that he believed that cancer cells ferment glucose in the presence of oxygen. And that was the hallmark of cancer. And if we can go into the hallmarks, it is a hallmark of cancer. But there's another component that if you're missing it, you might get misled and you might be confused. And the cancer cell or the human cell or the human stem cell, immune cells, there are other cells besides cancer cells that actually, they will ferment glucose. And I would argue glutamine in the presence of oxygen at certain times. And it's effectively physiological. I believe if you couldn't do that, it would be very difficult to repopulate your cells, for example, mm-hmm. which happens all the time. You've got your intestine, you've got like a brand new intestine after every week. Or at least that's what I'm told. So it's hard. It's hard to believe. But so here, if you look on the left-hand side, you see lactate. To me, of course, you see lactate go up. This is a graded exercise test, not the low and slow. But on the right-hand side, you can see this. It's cool. They like these plots. They literally have like all those dots. Those are all like the riders. So not only do they show you like the graph, I like that when they show all the individuals instead of just like here's your standard error. But you can see they bunch up and it's clearly high. The 4 asterisks is probably like a p-value that I'm sure everybody would die for, right? P less than probably 0.001, something like that. I actually have it down there, but not important. But anyway, I'm looking at this and I'm thinking, when you get to like exhaustion, I think it stands to reason that not only are you provisioning ATP from glycolysis, more of it from glycolysis. And I would argue it's very weird, like aerobic glycolysis, glucose fermentation, is that when you do a VO2 max test, that's like volume of oxygen consumed, right? Is that I think people fail to recognize that you're going to like, I'm going to kill myself intensity.
Mm-hmm.
And so you're actually getting energy from non-oxidative sources. In effect, I believe, and because whatever your mitochondrial capacity is, your oxphos, that's topped off. So what can you do? And what we can do is we can also provision ATP from glycolysis alone if, again, this is like all happening all the time. So when you say aerobic glycolysis, for example, aerobic glycolysis is like we always do aerobic glycolysis. If we're running glucose all the way through to the electron transport chain, it starts with aerobic glycolysis. So Warburg himself just said like these are going to be a nightmare because it's going to confuse people, and it did. So in this case, we've also got this other source of fuel, glutamine, and it goes to succinate. And they do speculate. I think I have some notes here, but okay. And this is the aerobic field test. So it's the same graph. And then you can see here lactate, succinate. They still saw like a little rise in succinate, and in lactate, it was actually kind of mixed, but considerably lower than the other one, which you would expect with the relatively low intensity. We'll skip that one. I'll just go to the succinate here. So this is individual cyclist analysis. This is like stage 6, and you got this hilly action here. This is the penultimate stage of the race too. So I don't know if any metals were, potential leaders were at risk here. These are they looked at 5 cyclists. And they studied how fast did they go, what was their speed. So this is based on performance, but we can kind of skip this. We can look here at, let's see. This is 6 metabolite correlates with average speed. And if you look at the bottom of the left-hand side, these are inversely correlated. So what this means is the higher the succinate, relatively speaking, the lower the average speed. The lower the succinate, the higher the average speed, which I suspect if you looked at lactate, it would be similar. So if you're able to buffer lactate and you look at your blood and it shows up lower, you're relatively fit, or you're able to push harder at whatever the intensity is. But again, here on the right-hand side, you can see the succinate is rising pretty substantially. And post S1, S4, S6, those are the different stages. And the stage 1 was particularly intense. And you can see the succinate is very high. So here we go. And then they're saying here, they're basically pointing out that, oh, here's succinate accumulation. Here's the succinate levels. And they're about even with lactate. We talk about lactate all the time as far as this is a marker of a threshold that you're going to hit, and that when you get to, when you cap off your intensity. And so this is what I have here with succinate, but effectively a cancer cell, our cells, our heart cells, like our cells are doing this all the time, but I think it's a matter of degree. So you have cancer cells, but they do a lot of it. And so you'll actually see it show up. Like in this case, if you're talking about exercise, it'll actually show up in your blood. You'll see succinate, you'll see lactate. But glutamine is a fermentable fuel. And so if we're talking about cancer and the idea that if cancer is a metabolic disease and the bumper sticker is damage or impaired or insufficient respiration, meaning that oxphos is somehow impaired or insufficient to get the job done to produce enough ATP, there's compensatory fermentation. And what Warburg believed, and what I think most people believe, if they even just believe or just want to like frame the theory, they think, well, the compensatory fermentation is through glucose. and then you blow out lactate. But there's also glutamine, and you blow out succinate. And so it's another fuel, and it's a fuel that's inside the mitochondria, which we don't have to get into this, but there's some fascinating stuff about that. So that's like a little bit on the succinate.
There we go. So essentially, mitochondrial capacity is, in these athletes, they're maxing out in these experiments. They're maxing out the capacity of the mitochondria using intensity. And basically, for something like a cancer cell, the argument is that the capacity of the mitochondria is being maxed out because its capacity is lower. And because you're reaching that capacity earlier on, it's tapping into either glucose or glutamine as these alternative sources of fuel that, and then in the measurements, they're measuring lactate, which is the byproduct of ATP production via the glucose pathway, and then succinate is the byproduct of energy production via the glutamine pathway. So they're measuring these things, and they see these things rise in cancer cells.
Yes, I would say when they look, they often don't look at the succinate. Obviously, Tom's looking, and he sees that. And so they actually did an experiment where they basically took away the non-fermentable fuel. So they took away the fatty acids, although glucose is fermentable and non-fermentable. A lot of these are they can do both.
Right.
But they basically, the typical thing that you'll see is that you can take away the oxphos fuel, and the cancer cells they'll run on glucose, and they'll blow out a lot of lactate. And so it looks like they'll ferment glucose happily. Their experiment, they pulled that out, and they said they took the glucose away, but they still had glutamine available. And if you look at any, you won't see glutamine. Oftentimes you won't even see it in the methods of papers, but the typical prep, I think it's called DMEM. It's like Eagle Prep. We use this thing. So they prep like their Petri dish, whatever they're doing in cells. It contains both glucose and glutamine. And so there's people talk about cancer cells being glucose addicted. And now it's like very in fashion to say cancer cells are also glutamine addicted. So they not only, they did one experiment, they pulled that out. They pulled out the glucose, and then I think they gave them the cells, they treated them with cyanide. And they're able to still sort of plug away.
Wow.
With glutamine. Well,
because the cyanide acts, the cyanide is a poison that acts directly on the electron transport chain.
Yes.
Right.
Cytochrome C, which is like the penultimate step sort of in the electron transport chain. Cytochrome C usually carries the electrons to complex 4, and then that's where oxygen picks it up and gets turned into water. And so you block that, you're effectively you're blocking ATP synthesis via the electron transport chain.
Which is the only way that you can get it via fatty acids. But with glutamine and with glucose, you don't need to go that route. You can get the glutamine energy from the Krebs cycle, and you can get the glucose energy in the cytosol.
Yes. Yep.
Very good.
So if you don't think about it in cancer terms, glucose and glutamine are like these beautiful fuels in a way because of that. I think it's like we sometimes throw around the world metabolic flexibility, right? It's like it's a fuel that our mitochondria can use. It can use it for the amino acids it can use, and then the glucose it can go either way. And under whatever certain circumstances, it can flip back and forth. It always flips, kind of does both, but it can rely on one over the other in order to basically survive.
And so that's why when you start researching into Seafried stuff, and I'll shut up so that you can keep going here, Bob, in a second. But that you start to see the reasoning behind Seafried's proposed cancer diet protocol. It's not only reduced in carbohydrates, but it's also reduced in protein intake. And really, it's a low calorie ketogenic diet, but you're basically eating exclusively fat because the glutamine would be coming from the protein sources.
Yeah. Well, Tom is of the opinion, I don't think I disagree with him though, is that he just says like, you can't, it's very difficult to lower your circulating glutamine concentrations through diet. Which I think is like generally probably true, although I don't think we've looked at in the context of what he's doing actually with, say, ketogenic metabolic therapy. And there is a rabbit hole that I went down with exercise, and there does appear to be something where there's actually a hypothesis that got dropped for some reason, but it was like in the 1970s, and I've like commandeered it. I actually thought of it, and then I realized, oh shit, somebody thought of this like before I was born. They call it like the glutamine steal hypothesis. And so that hypothesis is that the cancer cells, particularly the metastatic cells, we talk about cancer cachexia and how horrible it is. This is another one of my hypotheses, is that I think most of that, like a lot of that cachexia, is that phenotype that is being driven by glutamine being utilized by the cancer cell. And where is it getting it from? The biggest source of glutamine that you have is in your skeletal muscle. Like that's effectively, it's almost like your liver for glucose. Although even your muscle actually contains probably more glucose than your liver. And so your muscle is like this, actually the storage form of, it's holding the glucose and the glutamine. And so the hypothesis is that the cancer cell effectively basically steals that glutamine from the skeletal muscle. Is that normally the skeletal muscle probably throws some out into the bloodstream. It's constantly taking some back in. But now you have like this other source. It's not like I would say like you don't have like a placenta and a fetus, but it is like you've got cancer cells and you have like competition. And so some of the studies, some of them more recent, but there's some older ones as well, that they look at exercise, and there's one from like 2019, and they look at, this is in like rats and mice. And so they're doing running on a treadmill, which is actually fairly intense for them, is that the muscle tends to hang on to the glutamine, which would also make sense if you were growing muscle, is that it becomes stingier. And the tumor cells actually didn't, their growth was inhibited. And their growth was probably inhibited, these people through their like study, is that it was inhibited because they had less glutamine as a source of fuel. So that is one thing that I think for cancer treatment, one of the issues is I think a lot of times if you're a healthy person and you exercise regularly, it's very like these are, I don't know, I was going to say it's easier to take a drug, but arguably sometimes in cancer, it's not like people would actually opt for, no, actually stick me on Satan's tricycle instead. And I'll do that as a treatment, or at least like as an adjunct. But I think there's some stuff that's pretty compelling as far as exercise could be a way to at least slow the progression, and particularly for metastatic cancer, which is, again, the numbers are weird, but statistics are like 95% of all cancer deaths is metastatic cancer. So like that's really what we care about, is whether this local cancer takes off and invades other tissues.
Mm-hmm. Mm-hmm. Okay, very cool. So jumping in here, you see you got the title page up.
Yeah, so I've got like backgrounds. I have the cancer epidemic, and then I have I can go through, I'll do this very quickly, but this is like straight out of, I'd say, Tom's book too as well. He presented this way, but some of these statistics, they're just sort of, they're head scratchers for me. This is looking at like the last almost 15 years or so. And it's looking at new cases and new deaths. And each one of these are new cases and deaths per year. And so there's almost like 2,000 people a day dying from cancer, and 2 million people a year. So that would be even higher as far as cancer cases, which makes sense. So this is cancer deaths per year in the US. And this is crude. This is just like tally it up. And you're not supposed to do this because you need to age adjust, blah, blah, blah. But I just like want to put this thing in context that like 1880, the population was much smaller, but there are about 3,000 cases. There's 3,000 deaths from cancer. And we might not have been able to like, we weren't identifying as much cancer, et cetera. But this looks like, you know, this doesn't look like progress to me. But this one actually, this adjusts, still doesn't adjust. It doesn't adjust age adjust. It just adjusts for the population. So this actually does adjust for population growth over time, which you want to do because our population has grown substantially since the 1880s. You still see here, though, that it's grown pretty massively. And then you see like 1990 to 2000, there's like a peak, and then it starts to come down a little bit.
Yep.
And then this was from 2023. And so I looked at this, and I'm looking at it like cancer death rates fell 33% since 1991. Okay. It says partly due to advances in treatment, early detection, and less smoking. And I was trying to like come up with a fallacy, like when they do this, like this headline is effectively, it's directly from the report. So it's not like I'm not blaming the journalist, although I am in a way because they just sort of faithfully report whatever the authors do. But they're pointing out three things, right? Treatment, early detection, and less smoking. And so I called it like innocence by association. I don't know if that works. But the analogy I came up with is that if I've got a Porsche GT and two horses, and I say with these three things, I can generate 550 horsepower, right? But I didn't tell you that two horses generate 2 horsepower, and that Porsche, the GT, generates the other 548, right?
Yeah.
It's like less smoking, right? So like what's going on with less smoking? Before I do that, this is the 33% decrease they're talking about. You can't do absolute decrease. Like I'm doing a no-no here, but I did the age adjusted, all that other stuff. This actually looks like more, this looks better than if I did the other things. If I just looked at age specific, or if I just looked at it would still be, any way you slice it, the absolute decrease in terms of cancer mortality over this time, it's less than 0.1%. I've never seen them put anything like approaching something like this in their papers. And that is something that I'd say, don't trust the paper if they don't give you absolute risk. Don't even read the paper because you have to know the context. Because it's like, hey, it's like now 3 times stronger, whatever we're talking about, Gatorade, like stronger than what, right? So you go, cancer deaths decreased by 33%. Like 33%, what is that? Like what was it in 1991? What was it in 2020? Okay, this is directly from these same people. This is from 2026. This is death rates from lung cancer. And so you see this peak in 1991, and you have to always look at the rate, like what are we talking about here? Pretty substantial. So lung cancer is like a third of all cancer deaths. And there's a lot of cancers. It really is like lung cancer it's the BFD. It is the big deal. So when they tout like quitting smoking or reducing lung cancer rates, I'm like, great, thumbs up. They should probably focus on that instead of this other stuff where they're trying to basically pretend that screening and treatment is doing anything. But here, and then this is showing the trends in cigarette smoking.
Like if you go back to that last slide.
Yep.
Some of them it does look like there's, sort of a downward trend, but you look at prostate cancer, we're back to, it's the same as the 1930s. Yeah, it's like
they're age adjusted. They're not going down. They're age adjusted. And I had an article that I hope nobody read because it's excruciating. It's worse than this one. It gets into the nitty gritty, but what they do is when they have they should have it in here. It should say what they did, but rates are age adjusted to the 2000 US standard population. So what do? They have their data they have it all across. Right? Let's say that we're going to 2023 and they have their data from 2023. They've got their cancer deaths. They got all sites, and then they have all these different ones, right? Prostate, lung, colon, et cetera. And then they got, let's just say, the 1991, or even batshit crazy that it's like 1930 and they're standardizing it. I'll just say 1991, okay? They have the 1991, and they have like, they have 19 age groups, and so they do it by like 5-year age groups, so like age 5 to 9, age 10 to 14, and they look at the death rates for all of them, the actual death rates in 2023 and in 1991. And then they take the year 2000 population, which is mythical. Like it is a make believe. Let's make believe that both of these cohorts. I get angry, but there are contexts where it makes total sense. Imagine you're doing Florida and Texas after I say this, when you're comparing those two. But you make believe that your population in 2023 is the same as it was in 2000, and it is nowhere near the same. If there's an age pyramid where you see like the oldest at the top, they call it an age pyramid. It looks like a barrel now, because we do have a lot of, let's give ourselves credit maybe, is that we have a lot of people that are living to older ages. And in cancer, that is where most of the deaths happen. And so they are adjusting in 2023, where they have a lot of people in these older populations, and they're adjusting and pretending that those populations are smaller. And so they are undercounting on these age adjusted things. They're literally undercounting the number of the actual deaths. But it's weird because they go, those numbers on the left-hand side, even though they show a statistic, that is not a real number. I know it's kind of confusing. That is not a real rate of cancer death, because they're pegging it to a fantasy population. But they do this, and they, like in the paper, they probably won't say the peak was 85 or whatever it was, because it wasn't. That's an age-adjusted death rate. But when I found out, like when I started looking at like 2021, I did think, when you guys start talking about how you drop cancer death rates by a third, and you had 600,000 deaths in 2021, you should send a letter. You could do it at random, but when you do this statistical thing, your estimate works out to about 400,000 deaths. I said, you should send a letter to those 200,000 people and say, sorry, we didn't count your death because we're too busy having a parade about our 33% decline in cancer death rates. Like, I just think
is that the only motivation? Like, is there, what do they
so I
think
said
they're
Texas,
doing? Like,
so Texas and Florida, imagine that you're looking at Texas and Florida, the two states, and you want to compare them, not over time, but just say like in a snapshot in 2020, right?
yeah, at the
and you
same
look at
time.
the rates. You look at you just get the cancer death rate for both, and then you get the cancer death rate for the entire U.S. And you go, wow, Texas looks considerably lower than the U.S. average, and Florida looks much higher. And so you'd think like, well, what's going on? Like, do Floridians live in a toxic environment? Like, what's the carcinogen that they're exposed to, or whatever it is? But like, I think most people would know that the population in Florida skews where they have a lot of older individuals, and so they have more cancer deaths because they have more older people dying from cancer. Whereas in Texas, it tends to skew a little bit younger than the average population. And so if you just looked at the death rates, you wouldn't be able to like make heads or tails. So if you do take those two populations and you standardize it, like to whatever you standardize it to, probably to the overall US population, let's say, and then you can pretend that both of these populations have the same age distribution. So you literally age adjust for it. And then when you look at that, the rates are actually fairly simple, pretty much the same, fairly similar. And so actually when you do that, you would go, okay, this probably suggests that age has something to do with it. And then you can just look at the age specific rates, which I did in that paper, that article. And you can see that there's just more people at the at the higher age. And if you do want to track trends over time, you can, like, this is the way that you would have to do it in some ways. However, in a way, you would, if you just wanted the big number, here are the rates, the estimated cancer mortality rates for the entire US. And so how do they get that age-adjusted rate? I sort of said it. They take that standard population, and they have those 19 age groups, right? Age-specific death rates. And so I'm looking at it, and I go, why don't you just publish that? You can publish it for 1991, and you can publish it for 2020. And the articles say, these are from the American Cancer Society, National Cancer Institute. They say it's really cumbersome, which it is, and it's confusing to readers, which it probably is. Well, I would argue it probably is confusing. But relatively speaking, this isn't confusing because you don't understand what you're looking at. If you knew what you were looking at, you'd be bewildered at what you're looking at.
Right.
Because
like now that I know,
real rates. Right. Like,
yeah.
Those are fictitious rates on the left-hand side.
Yeah.
Right. But even though those are fictitious, the trend, if it was the 2000 population, you do get like a sense of what is the trend if the population stayed the same. But like, we're not living in that population. That's in part, like, that is why we have 650,000 cancer deaths or whatever it is this year. It's because we have a lot more people and we have older people, and that is something we need to deal with. And I would argue that stuff, that's like population level stuff. That is for epidemiologists to worry about. The age-specific death rates is like anybody who's like a reader or a person, they're like, oh, what's my age right now? What's my risk of death? Like, it's way more useful because you're actually looking at it like, okay, I can see, instead of like this number of the United States, you'd get more specific information. And so if you want to like cut it both ways, present both. You could present it this way, and then present it and then point it out the other way. And so there are completely justifiable ways to do it. I think it's just, there's a similar thing done with like randomized controlled trials. It's called intention to treat. You can use it, but it's like you have to be really explicit that you're using it and what it means and what you should and shouldn't interpret from the data. And they don't really do that. They usually say it's discussed elsewhere, and they provide a citation, and you can go search for it. They don't actually explain it.
Yeah. And if anyone wants a rant on intention to treat, just go to December's Journal Club where we covered the testosterone papers.
Oh yeah.
Yeah.
I got into that, huh?
Yeah.
Okay. So this is 1991. So they look at percent of all cancers. Look, where the cancer deaths, where they come from. And in men, so men are better at smoking. They do more smoking, I would say. So that's the hypothesis here. It's probably pretty strong at this point. Is that men did more smoking, and they also did more smoking. I don't have to go back to it, but they hit their peak earlier than women. But anyway, these are from 1991, and you've got lung cancers accounting for 34% and 21%. We talk about like absolute and relative. These are relative numbers, but they are absolutely big time because lung cancer, we're talking about total cancer deaths. So you've got 34 and 21. This is 2026, and you've got 19% and 21%. So if there's anything that I would look at with this, I would look at this and say, this is progress. And we can make more progress, probably, if more people didn't smoke. And so this is 2026. This is a whole nother ball of wax. So try to keep it brief. But this is literally last month. So the context here, so all that data, it's great stuff. The American Cancer Society, every year, they publish a paper in their own journal, which is a high impact journal, their own journal. I think it's called Cancer. And so you'll just see cancer statistics, comma, 2026. And that comes out in January. And you think like, I always think like, how do you have deaths and cases in 2026? Because they estimate them. So, but they do go back, I think, and at least check them. So the big thing here, at least, the thing that they promoted was that they hit a new milestone as far as 5-year survival, if you guys are familiar with, from the time of diagnosis with cancer, supposedly, what's the likelihood you'll be alive after 5 years? It's a little bit different than that. It's relative survival. So it's what is the likelihood of your survival in 5 years compared to a cohort that ideally would be like your twin that does not have cancer. And so that does make sense if you're talking about you're 85 years old, that's when you're diagnosed with a cancer. Your actual life expectancy is less than 5 years without the cancer. And so they compare with cancer, without the cancer. Although that, again, I think is very confusing because you'll look at something like a glioblastoma and somebody who's in that cohort, like a cohort of people aged 80 to 85. And if you see a 5-year survival of 5%, I think most people assume, oh, well, 5% of them made it five years past their diagnosis. That's not technically true. It actually could be far less than that, but it's compared to 85-year-olds without the cancer. So here, it's just like this quote. This is Rebecca Siegel. She's the one she's the first author on, I don't know, the 1991 was Boring, some person named Boring. And now it's Siegel. I think she's the chief scientific something. I'm going to get it wrong, but she's the bigwig scientific person there. So this is where the quote's coming from. And she says it takes decades for research to understand and develop these more effective treatments. And now we're seeing the fruits of those investments. This is our buddy Gerd Gigerenzer. All right. This is in 2013. He cites Gilbert Welch, this guy in the year 2000, who
he's a statistician, right? Gerd is a statistician.
Yes. Yep. Senior research scientist, Max Planck Institute. He does a lot of cool studies, and he does in this paper too. He does this thing too. He'll do like surveys and say like, "Hey, do any of you guys know what a p-value is? Like, what do you think it means? Do you think it means this, this, this?" And he'll ask like PhDs, and he'll collect the data, and he'll go, 10% of PhDs got the right answer.
Right.
And in this case, he does that thing too. So on the left-hand side, you got Giuliani. This was in 2007. And so he was talking about how he got prostate cancer and he survived it. And he goes, "Thank God I was cured of it." He goes, in the United States, 82% chance of surviving, like basically surviving if you get prostate cancer. And then he said, "How about England? Only 44%. And he points out under socialized medicine. So socialized bad. And then, so Gerd asked the question, why is an increase in survival from 44% to 82% not evidence that screening saves lives? And so the first one is lead time bias. Earlier detection implies at the time of diagnosis, blah, blah, blah. I've got a good slide on this. Basically, you start the clock earlier. Like you live longer knowing that you have the cancer now if you get screened earlier. That's one bias. The second one is this thing called overdiagnosis. And Emily and I, we argue with ourselves, but we agree with each other. How are you overdiagnosing something when you're properly diagnosing it? And sort of it's like almost like a statistical term, but by definition, you're screening cancer, but the cancer's a nothing burger. And so it won't progress to cause symptoms or death. But what happens when you screen and you have a positive test? You treat it because we don't know if that's a nothing burger or if that's an aggressive metastatic cancer in the waiting. It's going to drop you dead. So what happens? We treat. So overdiagnosis kind of goes hand in hand with this thing called overtreatment. And so overtreatment is like we're treating somebody for a cancer that shouldn't be treated for cancer, but we don't know that we don't know that we're overtreating them in the bargain, although we can find out, through some studies. So in the U.S., prostate cancer, we had the American Cancer Society, for example, we have guidelines for cancer screening. And in the US, we have a larger proportion of men being screened for prostate cancer. And it's something like, it's actually not that, it's similar for breast cancer, at least in one study or a couple of studies, as it is for prostate cancer. That if you look at like what decade you are in your age, that is the likelihood that if you die and they do an autopsy, that's the likelihood that you have that cancer. So in breast cancer, women in their 40s and 50s, they did autopsies, and about 40 to 50% of them had breast cancer. And this is like they weren't diagnosed. I mean, they were diagnosed on autopsy, which is, it's an effective way to diagnose, but I wouldn't recommend it, if you're actually alive.
Mm-hmm.
But in prostate cancer, it's similar. It's if you're 80, when you get to age 80 or 90 or something like that, it's conservatively more probably than not. Like if you got somebody to root around in there, or if you die and you want to find out, or you're somebody in your family dies and you want to find out, more probable than not that person has prostate cancer.
Right.
So
Gerd Gigerenzer, by the way, Gerd Gigerenzer is a psychologist. I just looked it up, but he researches inductive reasoning and reasoning under uncertain conditions. So that's why he's all about this stuff.
So this is lead time bias in a nice graph. On the left-hand side, you've got biological onset of disease, right? Like we can look at these people, crystal ball. We know that transform cell, number one, boom, that's when they got the disease. On the top, now we have a screening program, and we're looking, right? Like every annual checkup, we've got a good way to look at whether you have the particular disease, in this case, cancer. Usual practice on the bottom, let's just say like, we don't screen for prostate cancer. Then in both cases, let's say that this person died of prostate cancer on the top and the bottom. They both survived the same amount of time from the onset of their disease. But the person on the top, the perceived survival is going to be longer.
Mm-hmm.
That's the lead time. So they have a longer lead time. And so the clock starts earlier on the bottom. This almost assuredly, with the cancer cases, when they talk about relative survival and it going to 70% for most of these cancers, this is almost assuredly the reason. And so for like Siegel to say better treatments, doesn't even mention screening in that quote. It's like, you got to be more careful than that. Or I don't know, maybe she is being careful, and that's why, who knows? So 1991, I try to look at 2025. The American Cancer Society has a nice website. If you type in like ACS, lifetime risk of dying cancer, you'll find for all cancers, the probability that you're going to die from any cancer, which in men and women, it's actually getting closer. Maybe that's the lung cancer, the lung cancer reduction in men. It's about 1 in 6, 1 in 5, 1 in 6 for any cancer. And so that's readily available. I couldn't find, 1991 is where so the thing that they do is they take the peak, they take the height of the worst too, is what they tend to do, and then compare everything to the worst, and then look at their numbers. So I tried to do that. I looked at the most recent, I don't know, I got some help from ChatGPT, doing like mortality hazard estimates. I did get the data from SEER. It's like the same place that they get the data from. In 1991, it was about 22%. And so this is your lifetime risk of dying in 1991, 22%. I mean, you could argue like that those are real differences. To me, I'm looking at it like it's probably around 20%. And the thing that they're not talking about, like I think that they should talk about it more, is like these are just averages, and that there are things that you can do, even though my papers on hereditary cancer syndromes, is that the environmental factors are really important. And there are things you can do that are likely going to reduce your risk. And so it's not just like, this is your probability. But anyway, there it is.
Well, I think your papers on hereditary cancer syndromes, but when you read it, the conclusion is that, hopefully if you read it and agree that there's a strong likelihood that cancer is a mitochondrial metabolic, or that's at the root of its formation and the basis of the cancer, then you quickly come to the conclusion that there's a lot I can do to prevent
Yes.
cancer in myself.
Yeah.
Right.
Yeah. I sort of champion the person, even though I also kind of cringe, which is that it's Mary Claire King, who she was at a company, whatever, like there's all drama behind it. But ultimately she call her the co-discoverer, pretty much discovered BRCA1, and then BRCA2 is later, I think. But BRCA1, she named BRCA1. And BRCA stands for breast cancer. That's why I cringe.
So
It's almost like
BRCA1 and BRCA2 are the hereditary genes associated with that line of breast cancer.
Breast and ovarian cancer.
Both.
And somatic mutations as well. But so she coined it BRCA1, and then she had a paper like in 2006, and she started. She could get data, historical data. I think back to like prior to 1950, and then she looked at like more recent, and she looked at like BRCA1 carriers, and in the 1940s and 1950s, substantially lower. The lifetime, the quote unquote penetrance. Penetrance is like a genetic term. It's like Huntington's disease is a great example of that. Supposedly that's 100% penetrant. Huntington's disease, you're going to get it if you live long enough.
I think you might define it in your paper. I think you talk about that pretty early on as penetrance, and you refer to that term quite a bit through the paper.
That's the jargon, but I think it can be confusing because the penetrance can be variable. And in this case, Mary Claire King, the person who just identified / discovered BRCA1, pointed out, she goes, look at these two vastly different rates. She goes, clearly something outside of genetics is going on. And then environmental factors are probably very important, something to that effect. And so she was pointing this out, what is that, scary, 20 years ago, 20 to 30 years ago, saying there is something it's not, so I think it's like 50, depends, right? 55, 65% of women with BRCA1. It's going to depend. Lifetime risk. So lifetime risk of getting breast cancer versus dying from breast cancer. She was pointing out that there are things you can do to lower your risk. It's not only is it not a death sentence because it's not completely penetrant, but there are things that you can do to affect it. So, yeah.
Were there cases at one point of women getting preemptive mastectomies when they found out that they had the BRCA gene? Is that a real thing, or am I imagining things?
Yeah, that's a real thing. That was there's a famous one, Angelina Jolie. She wrote an editorial about it in the New York Times, and she had a double mastectomy.
Okay. Because she found out she had either BRCA1 or BRCA2.
I think I know exactly what she had, because it was, I don't, what would I call it? It was very specific. When she said, my doctors looked at my family history, whatever, and they say, I forget what the number is. They say that I have a 84% chance of getting breast cancer. And I was like, 84%. Right? That's very specific, right? So I went back and I looked at the paper that's cited the most. Is that there's like a population paper from, it's like the mid 1990s, I think. And they looked at families with the BRCA1 and their rates of breast cancer.
Mm-hmm.
And so whether it was her doctor or whether she did the research, it was, I would bet on it, at least. I would bet that is where that information is coming from. And if you look at there's another thing called ascertainment bias. So when you look at populations that families with how do they find the BRCA1? They have to find families with a remarkably high rate of breast cancer in the first place. And so there can be other factors, or it can even be purely genetic, that the BRCA1, the particular kind of mutation that they have, either way, you're selecting the most vulnerable population, and you don't know there might be a graveyard, so to speak, of people with the BRCA1 mutation that are asymptomatic, and you just don't know that they're hanging around. So more recently, they have like the UK Biobank, and they have these other things where they put their blood, their saliva, all that stuff, and they're getting some of these population tests, and some of these penetrance numbers are getting like blown to pieces because they were showed it. Like one example I use, I think, is MEN2, multiple endocrine neoplasia, type 2. And for one particular like mutation, they say the rate, like the lifetime risk is something like, I want to say 90%. And in one of these more recent studies, they were able to get call it a pure population of people that didn't have a family history. And it effectively estimated, they said conservatively, it was like the lifetime risk was like 3%. Like 3% versus 90%. And so with BRCA1, even like the more recent studies, it's not a nothing burger. It certainly looks like it's higher than if you don't have it in the United States. Although I do think there's a genetic environmental interaction with this stuff. But those early papers, it can basically go no other way than those penetrance numbers will be on the decline from then on if you're able to get a broader swath of the population. And so those are the highest of the high estimates you could possibly get. And so I don't know, it concerned me when I saw that. But yes, and so a lot of these, they call it prophylactic surgery. So there's that MEN2, there's a particular kind that it hits your thyroid. It's called medullary thyroid cancer. And this was the one I found probably the most compelling, but it had so little information because it's so exceedingly rare, is that I think the American, they call it the thyroid cancer society, let's say. I hope I got that right. They recommend prophylactic surgery for basically infants to remove the thyroid because like the data that they're looking at is like, this kid's going to die by the age of 1 if we don't do this. And so that's what happens. So what happens with a lot of these hereditary cancer syndromes is that when we talk about screening, and you talk about like lung cancer screening, for example, it's recommended for a certain population. It's recommended like when you're older and now it's like based on pack years. But breast cancer, for example, now I think it's in women 40, but it won't start in asymptomatic women until a particular age because there is risk of screening. And so they're trying to weigh the risks and the benefits. When they weigh the risks and the benefits, when they look at these populations, they see far greater risk. And so when they look at like a prophylactic surgery, there is certainly risk associated with prophylactic surgeries where you're removing organs and tissues, but they're also weighing that against what they see as far higher risk. So yeah, there's that does happen, and it still happens.
Yeah. So, I mean, so like the implications of this research you've done is huge. It's not just theoretical. There's real people who fall victim to, sort of old views and misunderstandings of statistics or not the whole picture necessarily. And so this is, it's another piece to the puzzle to hopefully give people a better outlook on what's actually going on.
Yeah, and I think that one's a difficult issue because you don't have to look at cancer as a metabolic disease or a genetic disease. You just have to look at the way that like the populations are drawn and statistics are drawn.
Yeah, the statistical analysis is really a big one.
What's going to happen is you're going to underestimate, you're going to overestimate basically the severity of the disease.
But even so, I mean, the mitochondrial, the metabolic versus somatic theory matters too, though, because if you believe that the primary factor is the genetics, and you can't change your genetics, then you might lead yourself to believe that it's inevitable, like it's a death sentence, and I can't do anything about it. But if you have the same likelihood of dying of a certain cancer, and you know that it's a metabolic disease, then that might change your perspective slightly, thinking that, oh, well, I can make these changes in my life, in my environment, to potentially lower my chances by, X amount. The outlook changes a little bit too, depending on those two views.
Oh, absolutely. There's a great case or a story I read was Alzheimer's disease. You've got what do you have? You have like three supposedly deterministic genes in Alzheimer's disease. Most people don't have those, but the one that probably most people know is ApoE4. And the guy that discovered ApoE4, this guy Alan Roses, I think he may have passed away recently. He discovered ApoE4, and he would talk in his talks. He would say, "In my laboratory, if I find that one of my postdocs, one of my researchers themselves got a genetic test to see if they had ApoE4 or not, I would kick them out of my lab." And he would do that because he said, there's nothing they can do about it. So all they're doing is creating this worry that there's nothing that they can do about it.
Right.
And I think somebody could look at that from a different perspective and say, there's a hell of a lot that you can do about it. Like if you look at the nature of ApoE4 and the evolutionary history of it. But I don't know if this is a funnier story, but he also discovered later on, it's called TOM40, TOMM40. And he hung on that more than ApoE4. And it's kind of like it sits in the same genetic locus, et cetera. But TOM40 it's called translocase of the outer mitochondrial membrane, 40. It was a mitochondrial component. And what he was pointing out, he would do in his talk, I thought it was kind of funny. He goes, that Roses guy was full of shit because he was talking about like ApoE4. He was saying ApoE4. No, it wasn't. He said that it was it's sitting in that region. And he was arguing that it was this TOM40. But I don't know if he would just think, well, that's genetic too. So absolutely, it depends on your outlook. But certainly, I think even a lot of these people are sort of in the weeds. This is genetic. This is hereditary cancer syndrome. They know that the penetrance numbers can change like remarkably over time, just based on the data that they're drawing, and so that they know that the penetrance can be variable. The problem is, this is what I think part of the problem is that there isn't like the first do no harm in medicine. It's very difficult to do. Like don't just do something, stand there. That should probably be their motto in some cases. I think it's really hard to do. It's like you diagnose somebody with a disease that could be devastating, right? What's the precautionary, like what's the safe thing to do? And I think most people think like the safe thing to do is, it's like, was it duck and cover or whatever? What was the lockdown? The lockdown was like, it's part of that like rationale, right? Let's err on the safe side. And you think there might be unintended consequences of doing that, of erring on the safe side. And clearly, Peter Goche is known for this. He co-founded the Cochrane Collaboration, and he was somebody. It was like 2011. He wrote a book on mammography that I realized like I'm probably not taking crazy pills, because some of the stuff that I was seeing, he had already pointed this stuff out. And Vinay Prasad had done a lot of stuff on it too as well, and that's somebody that I followed on this stuff. And he said like the risks are real of screening people and diagnosing cancer in people that otherwise would not be diagnosed of breast cancer. But it's very difficult because it's sort of like I don't know what the bias is, but you have all these. I just drove past a hospital near me, and it says mammograms save lives. Huge banner, right? And when a woman gets a mammogram and they find a breast cancer and she's treated, and she's free and clear, and she rings the bell, she thinks like that mammogram saved my life. I think like, what if you didn't get the mammogram? We don't get you. Like if I'm the person that's I'm not a proponent of mammograms, or I don't think that she should have got one.
Mm-hmm.
She can't tell me if that mammogram, not getting the mammogram saved her life. But it is true that there are some people that die as a result of overdiagnosis and overtreatment. And that's something that Gautier was effectively like he was fighting for. He's at the Cochrane collaboration. He seems like a logical person. And he's like, why am I being shut out? And he was like, I underestimated like the power of some of this stuff. But
yeah, I think the same conversation is had around prostate cancer screening, and
yeah, it's in this 2026 paper too as well. Yeah. It almost looks like they're sort of slapping. There's so the American Cancer Society has recommendations for when people should get screened, healthy people, for all the various cancers. And their recommendations are different than what's called the United States Preventive Service Task Force, the USPSTF. They changed, I don't know if they changed the recommendations, but around 2007 to 2009, they told people not to get, like whatever the age groups, they didn't recommend screening for prostate cancer. And so in the paper, it's looking like in the cases, it looks like the cases were going up, and they were concerned about the number of cases going up. And they're thinking like, this could be a problem. But meanwhile, the cancer mortality rate didn't go up. It actually went down, I think, over the same time. But there are different camps. They're all they're not in lockstep. But the USPSTF, I don't think that they're perfect by any means. But I do remember when I was doing work for cancer screening, things like that, they do take out some things that I would want to take out if I just want to know how effective is screening. Like some of it is like cost, whether it's cost prohibitive and things like that. And I think like that is really important, that if you have to pay out of pocket, it's going to certainly impact whether you get it or not. But I don't want that to cloud whether, does this thing work really well or does it not? And that's something that they do.
So I told you I'd keep you on track with the time frame here. So we're past the first period. We got our first hour in. And it looks like we're getting into a little bit past the statistical stuff and the introduction of the history of statistics of cancer in the population, and we're getting into more the cellular biology now.
Yeah, we're getting to the two theories. And I'll say like with the paper, if you read like the introduction and discussion, or I think if we go through that, and I can kind of briefly look at them in totality, because a lot of it is alphabet soup. It's just for all these different hereditary cancer syndromes, but it's all alphabet soup that points in the same direction. All of these things seem to go in the same direction, where they either, most of them seem to impair mitochondrial function in the form of oxphos or respiration. Well, I'll explain that probably in a second. And or they promote compensatory respiration or compensatory fermentation. But here we're looking at like the cell, we're looking at the nucleus of the cell. This is where we think, if we're a somatic mutation theory proponent, this is where cancer comes from, in the nucleus. But you also have, so that was in the textbooks, you also had mitochondria hanging out there, right? Kind of lining the cell. So you have many mitochondria for a given cell in some cases. Red blood cells, you don't have any mitochondria. You also don't have a nucleus. And that also makes up like 85% of all your cells. And so when people say like you've got 30 trillion cells, all of them have mitochondria. I was guilty of that too. I'm like, well, there's 25 trillion of those cells are red blood cells, and they don't have mitochondria and they don't have a nucleus. But this is a static look. This is a textbook look. Mitochondria is mitose and chondria, which is, I think mitose is thread. Chondria is like grain or granule. Okay. So it's like a bean. That's what we see, like a little bean. That's what it is.
But hold on there for a second though. It like the importance of the red blood cell, like just so people know, is to deliver the oxygen to the mitochondria and all the other cells. Right. So you got those mitochondria less red blood cells, but really their main purpose is to support all the cells that do have the mitochondria. It's a fascinating rabbit hole, because I thought that makes sense, that you don't want the thing that's delivering oxygen to be consuming a lot of oxygen in order to stay alive. Right? It's the red
red blood cell steal hypothesis, but that's what they're there for. I looked at birds. Birds are like, they're the ones you talk about, like chasing perfection in terms of mitochondria or whatever.
Mm-hmm.
They got to fly, like they have flight, right? Their red blood cells do have mitochondria.
They have them.
I was like, I was like, wow, that's interesting, right? So I think there's got to be a there. There's got to be something you can glean from that, but
yeah. Okay, for another day.
They have it, we don't. Yeah. So this is through a microscope. You're looking at, like, in the green, that's all mitochondria, like spaghetti networks that surround the nucleus. So it's not just like these beans that like sit in this gel, right? These suckers are very active, and they are talking to the nucleus, because the nucleus basically stole most of their genome. And so they actually, a lot of it is like they're knocking on the door for what, like, the proteins that they need oftentimes. The only ones that they retained, and it's like something like in number of like 70 genes that encode proteins, they're all in complex 1, complex 3, and complex 4, and those are all the proton pumping complexes of oxphos. Probably a pretty important reason why they hung on to those. This one, I think we actually can get a little animation.
Wow, that's
so the
cool.
left-hand side is, and so I look at this. This is totally teleological. The nucleus on the left-hand side, the green, it could be, let's say it's doing lots of stuff. I just always think about, like, who's calling the shots here, right? Like, you've got the nucleus, which is like, you've got the DNA, which is a blueprint. And then now we know that there's epigenetic signaling, and that the nucleus is effectively can be signaled. It's called retrograde response. We'll get to this. Retrograde response is what's called when the mitochondria signal something to the nucleus and says, hey, fill in the blank. I need this, I need that. And then the right-hand side just sort of shows you these things are like expanding, contracting, they're fusing and fizzing. So they're breaking off, and they're joining forces depending on what's going on. It's just like, it's like wildly dynamic what's going on with the mitochondria. And then when we go to somatic mutation theory, this is the National Cancer Institute. You go to their website, what is cancer? And how does cancer develop? And you can see cancer is a genetic disease. That's their line. So here we are with, this is the somatic mutation theory, is that it's not really like a theory, I would say. You could call it like you can call it, people say it's like a dogma. I guess that's probably what it is. People don't even think of it as a theory. It just is.
It just
cancer is.
Yeah. Yeah.
It's
a fact.
Right. And so they it's not like they don't even argue it anymore. That's what it is. Okay. This is Paul Davies. He's a little snarky. I don't know if you're going to get it from him, but Paul Davies was one of these, this is fascinating. And I credit the National Cancer Institute for this. Like around 2000 to 2005, someone at the Cancer Institute said, like, we're not really making all that great progress. It seems like we're stalling here a little bit. We're stagnating. And so they tried to, like, hire outside sources that didn't know anything about cancer. And so one of these guys, they had these like astrophysicists, this guy Paul Davies and Charlie Lineweaver, and they brought them in. I forget what the project was called, but they brought them in to say, what do you guys think? And so they looked at it, and if you can see the title of this paper, the title of the article, this is What Scientific Idea is Ready for Retirement. You might be able to guess, but anyway, these guys have it's a hypothesis that I think, in a way, is very compelling, which is, it's called the atavistic hypothesis. And that it's cancer arises effectively because through our evolutionary history is that we can't get rid of cancer because it's recapitulating effectively embryogenesis. So what does he say? This is the idea of the somatic mutation theory. You have a cell and it serially accumulates genetic damage to get a mutation, and it reaches a point where basically it stops listening to the whole, the collective, and it acts on its own agenda. And one of the things too is it acquires distinct, like very specific hallmarks. And Hanahan and Weinberg, I think it was in 2000. Robert Weinberg, he writes, like, it's probably behind me, The Biology of Cancer. So if you want to get cancer from a genetic disease, you can read that book, or read these things too. But he and this guy Doug Hanahan tried to put together, like, cancer's crazy, complex, multifactorial. They throw out those words. Look, what do they have? Like, what do these cancer cells have in common? And so they listed the hallmarks of cancer. I have this number of citations, so I'll get to it. But so you have, like, all these random somatic mutations that actually seem to elicit this more specific phenotype. And what Davey says, I like this line actually here. He says that cancer with all these attendant hallmarks is considered to be reinvented de novo in each host organism, the result of a dream run of lucky genetic accidents. And so you get all these random mutations, and it's almost like winning the lottery, but not the lottery that you would want to win. And that's what this looks like. It's clonal expansion. And so you start with, like, this, you could have, like, a cell at the way top that would be the normal cell. And so you have the initial mutation in pink, and then you've got that mutation, those cells divide, and then picks up another mutation. That's the one in red. And all of these things basically have to do with cell cycle. So all the genetic view stuff, that looks like alphabet soup, but similar to I would say the mitochondrial metabolic thing, ultimately, like, you'll get to P10, all the Cowden syndrome, Lee-Fraumeni syndrome, P53. It either, like, helps with DNA repair, or it does something with the cell cycle. And so basically, like, it takes the brakes off, or it sticks the gas on. And so here's the hallmarks of cancer. And so this is actually, I think, a pretty good explanation of how it's looked at. You've got oncogenes, or proto-oncogenes, and tumor suppressor genes. And so you could have faulty breaks or overacceleration in a cell. The faulty breaks are, normally you have these tumor suppressor genes, like BRCA1. It's interesting because we say BRCA1 like that's a bad thing. We all have BRCA1 in called the natural or unmodified or mutated form. That's a good thing in terms of tumor suppression. Yes. If that has a mutation, most of these cancers that I went through, virtually all of them, they're tumor suppressor genes. So they're the so-called faulty breaks. The other one, overacceleration, is an oncogene, that MEN2 that I mentioned. The gene there is called RET. RET is when that thing is overexpressed, that promotes cancer. And so that is an oncogene. And that would be your foot's, like, stuck on the gas pedal. Mm-hmm. And then that analogy is you've got the mutations in tumor suppressors and oncogenes, and you've got cell division out of control. And then here, this is just the clonal expansion thing. It really is, they're saying, like, this is like Darwin in like ultra fast. So when we look at, like, Darwinian evolution, and it takes, who knows, hundreds of thousands, if not millions of years, where we start to see, people with a particular, ApoE4 is a great example. That ApoE4 was the ancestral allele or gene, is that we all had ApoE4. And so ApoE2 and ApoE3 came along later. Those were considered the minor alleles or mutations. And today, ApoE4 is now like the minor allele mutation. So these things can change over time. But this is like, we're looking at this thing in a cancer cell. It's happening very fast. There's some challenges here. These are some challenges to the SMT. We've got observations that we're finding cancer cells that they don't have gene mutations. So if there's no gene mutations in the somatic, that is a challenge to the somatic mutation theory. That should be pretty straightforward. And then you also have cancer driver mutations found in normal cells. So there's been a lot of recent work where they'll look at people and your skin cells, etc. They contain the driver mutations, yet there's no malignancy. Many carcinogens do not cause mutations. And then we have chimpanzees, which are very close to humans. And there's a rarity of cancer in this population. And in primitive populations, there is a rarity cancer, and then there's a nuclear transfer experiments. We can get into it in a little bit. This is looking at there's no mutations, and this is from Seafried. He says cancer cells with no mutation should not exist according to the SMT. It's correct. And in their papers, actually, I think it's Bert Vogelstein, he calls it dark matter because we don't see mutations. They're probably there, we just don't see them. This is cancer driver mutations found in normal cells. And so here, if cancer driver genes are the cause of cancer, then why do we have so many driver genes in people who don't have cancer, but they don't express it? Many carcinogens. So this paper is 2020. I found this kind of remarkable. They looked at 20. This is like looking at like the World Health Organization. They've got a huge list of like known carcinogens, group 1 carcinogens. These guys looked at like the ones that are known or suspected to cause cancer in humans, and they looked at it in mice where they also did some analysis where that's normally they recapitulated in human populations to see similar things happen. And in the 20 the carcinogens, only 3 looked like they had a mutational signature. So effectively, like three carcinogens were mutagenic, and then the other 17 were carcinogenic but not mutagenic. So they appeared to be causing cancer, but not through mutations, which again would not support the somatic mutation theory.
Yeah, very interesting.
This is so rarity of cancer in chimpanzees. This is Seafried again. He points out breast cancer has never been documented in a female chimpanzee, which is pretty remarkable. That's one that I would like try to go and check.
And they've got BRCA1, I'm assuming.
They have BRCA1, and that can be mutated. Yeah.
Right.
Okay. These are primitive populations, for lack of a better term. These are the Ake. I don't know if I'm saying that correctly. The dotted line, you can barely see it. That's the plausible rate, cancer death rate in wild Ake. And I think in part, the authors kind of like, they say plausible cancer death rates because the cancer death rates are so low. I think they assume that they must be missing some cancers. They're not diagnosing them. It must be there. And that's kind of prevalent in a lot of the historical look at a lot of these primitive populations. There's all these accounts. It says, so that even their discussion, they say, this rarity of cancer in primitive conditions is not a new thing and it's confirmed elsewhere. So it's confirmed in a lot of places. It's just a matter of like, the data isn't perfect, but it's been repeated throughout many different primitive populations that it's like a non-existent thing, at least in these populations or doctors that go to a particular population and look at it.
Mm-hmm.
And then this is the Succinate, very similar. And they say, note the river region has a cancer death rate of 0 over an exposure of more than 500 risk years. It's basically like, it doesn't exist, or maybe they don't get screened all the time. And maybe there are some deaths, but nobody's properly diagnosing them. It's possible, but these people go over, I think, and look, and oftentimes they'll confirm that they're not finding it. Now, the nuclear transfer experiments. So this is at the top, you've got on the left-hand side, if you transfer, you take a healthy cell, you take a healthy cell and you transfer the healthy cytoplasm and the healthy nucleus into another cell. And so you have that healthy cell. Healthy cells beget healthy cells, which should be pretty straightforward. Mm-hmm. And on the right-hand side, pretty straightforward too, tumor cells typically beget tumor cells. And then you have the transfer on C and D. On the left-hand side, you take the tumor nucleus and you implant it into a cell with a healthy cytoplasm. That's the left-hand side. The right-hand side is you take a tumor cytoplasm and you put that into a healthy nucleus. And so on the left-hand side, you see when it's a tumor nucleus and a healthy cytoplasm, those cells beget normal cells. On the right-hand side, when you have a healthy nucleus and a tumor cytoplasm, those cells beget tumor cells or death.
I mean, that's probably the most convincing and profound experiment for the argument that it's a metabolic theory. Who is the first person to do this experiment? And how many people have recreated it?
I want to say around 1950, maybe 51. Somebody named McKinnell probably did something similar to this. I don't know what the technology was or when that became available at the time. But there are various different experiments like this that they've been, I would say, reproduced and replicated. So some of them are like different situations. Some of them look at like frogs and tadpoles, and different variations. But it seems like the finding is relatively consistent, and you have more recent papers that look at this as well. I think I do have.
It's been done with human cells?
I don't know. Maybe. Not in humans. I would suspect they wouldn't implant these in humans, but they could use human cells.
No, but in vitro.
Yeah. I think so, but in this case, they're looking at, and this is Israel and Schaefer. This is UVM. These are, again, people that might have been around, might have been there when I was there, undergrad. I didn't know they existed. They did experiments here. And so what they did is, you could call it in vitro at the top. And so they had normal cells, normal rat liver cells, that they grew them in culture, and they like hundreds of generations. And ultimately they found from the same original cell, the same clone cell, they actually had what looked like a malignant cell. So they isolated that. And so they have this malignant cell from that normal cell top, of those normal cells. And they also had normal cells. And so then they implanted those in a rat. And so on the right-hand side, you see 12 out of 13, and that is, they implanted the malignant cells in a rat, and 12 out of the 13 went on to develop a tumor. On the left-hand side, 0 out of the 11, which probably fairly intuitive. I guess normal cells beget normal cells, cancer cells beget cancer cells, and if you rough up, they have techniques too to scrape them up, but they do it in both cases. And so then they have the malignant cell, the normal cell. And so then what they did is they extended this experiment, and now you're looking at the middle of this graph, and you have the red nucleus is from the malignant cells, and they put that into a normal cytoplasm, which is effectively what we just saw, basically. And then you have a green nucleus from the normal cells put into a tumor cytoplasm. So on the lower left-hand side, you've got the healthy cytoplasm and the tumor nucleus. And let's see what happens to the rats in this case. 1 out of 72 went on to develop a tumor. And on the right-hand side, where you have a healthy cytoplasm, a healthy nucleus and a malignant cytoplasm, virtually all, I think that's 97%, formed a tumor.
Wow.
So, I mean, there can be things wrong with the methodology and the experiments, but it certainly would lead you to believe that there's something going on outside of the nucleus that's really important here.
Mm-hmm.
I think it's difficult to just explain it away with other data, because I think this is pretty compelling. You would want to at least, I would think, refute this directly if you were somebody who's a proponent of the SMT. So mitochondria are abnormal in GBM. This is just good pictures from, this is one of Tom's colleagues. The left-hand side, you've got the normal mitochondria. It's got the invaginations. They're called cristae. You can sort of see there's your bean. And then on the right-hand side, they call it ghost mitochondria. And it's ghost mitochondria because you look inside and it looks like it's like hollowed out. And so you don't have those cristae. And the cristae in that surface area, that's where all the protons are being pumped. That's where all the oxphos is occurring.
GBM
amongst
is
other
glioblastoma.
Yes, which is it's like a grade 4 astrocytoma, which is like glial cells in your brain. It's particularly brutal. Not a great prognosis on that one. And then this is, sorry for the table here, but I just took it from Tom. Sorry, Tom. The mitochondrial abnormal, he went back and he looked at, this was in, I think 2020 he did this, and he looked back at all the different cancers, all the major cancers that basically account for the majority. I think he said 90% of all cancer deaths this represents, all these cancers. And he found abnormal mitochondria in all solid tissues and blood cancers. And then he also found, interestingly enough, on effectively that same groups of cancers, these lipid droplets, or cytoplasm lipid droplets as well. And these can be also, it's an indication that the cell may not be able to use those fatty acids for energy, and they're accumulating these lipid droplets. So this could be, in effect, this could be a biomarker for cancer as well, or a biomarker for impaired oxphos. Although there are other theories about why they would, in part, this is the sequestering of these lipid droplets is in part is like in these cancer cells, the mitochondria aren't gone, but if they are, let's just say they're messed up. So we talk about reactive oxygen species a lot, is that if they use this fuel quite a bit, they're going to blow off a lot of exhaust, like not physiological high amounts of ROS, like mutagenic levels of ROS. And so in a way, you could argue this is called a defense mechanism, is that they're sequestering these lipid droplets because when they burn, if and when they burn these things, they're done for, they're goners. Either apoptosis, necrosis, the fill in the blank type of either programmed or unprogrammed cell death in those cells. So this is the mitochondrial metabolic theory of cancer. So chronic insufficiency to cellular respiration, that's oxphos. So insufficient oxphos, and then compensatory fermentation. So lactate fermentation compensates for insufficient oxphos, and that's cancer cells ferment glutamine as well, even in the presence of oxygen. Enhanced fermentation, that is the common physiological malady of cancers, is how Tom puts it. And it came on long later, but it's called reprogrammed metabolism in the hallmarks of cancer. So this is a large figure that kind of shows this is what's going on in effect mechanistically with the origin of the MMT. We'll come back to this. I'll come back to that one. This is, Pete, you probably know this sucker well. Energy metabolism in normal cells. This is, generally speaking, where does most of the ATP come from? Comes from oxphos, about 90% of it. And then you got 5%. It's dependent on different cells. You got muscle cells. You even have type 1 fibers versus type 2, and they're different. But generally speaking, this is what's going on. And in biology, it's almost a safe bet that like everything's going on always. It's just at different degrees. But typically, it's predominantly most of the ATP is coming from oxphos. And then in cancer cells, what can happen is oxphos can be substantially diminished. And if that cancer cell isn't a dead cell, then there has to be a compensation, because no ATP, no life. It's almost like by definition, if that thing's alive, somehow it's getting ATP, and it's called the delta G of ATP hydrolysis, and it's within like this very fine range that cells have to maintain. And so if there's a perturbation that's extended, like it lights out effectively for the cell. And so what happens is, Warburg pointed this out in 1956 too. He said, you can't get cancer cell from a dead cell. And so he said, and there's probably a hell of a lot of dead cells. And there are a lot of cells that can't compensate. And so ultimately, like the ones that do are the ones that aren't dead, and they're the ones that are fermenting. And interestingly here, this is Tom's figure, but he's got mitochondrial substrate level phosphorylation at about 65%. So he thinks it's like it makes up a substantial portion of
that's a lot.
I think it's variable. I mean, I think it depends probably in part on fuel availability and the tumor microenvironment and things like that.
Mm-hmm.
I do suspect that more aggressive, when you're in the stages of metastatic cancer, you're probably looking at more MSLP. I just think because it's being drawn from you effectively. If that's being drawn from you, the phenotype is like the scary things that you see. You see cancer cachexia, and that is like it's a leading cause of metastatic death.
Mm-hmm. Yeah. MSLP being the glutamine.
Yes. So that's the MSLP is the mitochondrial substrate level phosphorylation. So that's call it the fermentation that's going on in the mitochondria. And then the CSLP is the cytosolic substrate level phosphorylation. And that effectively means like it's done at the substrate level. It's not done at this like amazingly wonderful thing that's pushing protons chemiosmotic.
Yeah, this like
Peter Mill
basically
Peter
this
Mitchell,
engine.
It came up with this hypothesis. People thought he was out of his mind about how we make ATP. But I guess he had the last laugh, sort of, on that one. He got the Nobel Prize, and he's got a great. It's called like I forget what it's called. It's called like the luncheon or something, and they give talks before they like accept their Nobel Prize, and he's just like, thanks to all the haters. It's funny, but he does. If he's being honest, though, he said like you really, like, you held me accountable, which is probably true. So here you have glycolysis, hemolysis. This is cancer cell growth, and this actually alludes to that. This is Derek Lee's paper, and that you have these two fuels, and they're sort of brilliant fuels in the way that these are great fuels through those called substrate level pathways. There are other pathways too that through glutamine, if you run glutamine the other way, or if you don't just run glutamine through the Krebs cycle, you can make building blocks and you can make energy with glutamine. With glucose, you can make building blocks and you can make energy. And so you can make them through both pathways. And so these things kind of probably compensate for one another as well in cells, in normal cells, in cancer cells, immune cells, et cetera. But you'd say it's like brilliantly awful in the case of cancer cells that won't seem to die. Brilliant in the way that we probably wouldn't be here as people. It just didn't exist in the first place. These are from Tom, but this is a mitochondrial like which one better explains the observations as the origin of cancer? The reliance on SLP is as the metabolic hallmark of cancer, and you have, this is more of a treatment thing, but simultaneous glucose and glutamine restriction under nutritional ketosis can manage cancers, and that's shown in, I would say that's shown in preclinical models, and I think I would love for us to look at, the population of interest, if that does, but looking at those trials. And so press pulse is this idea that you can restrict glucose. You don't have to eat glucose. We'll make all we need. And so you can bring those levels down to zero. Glutamine's called like conditionally essential, but if you are even, let's say like therapeutically, you're just going to deplete your levels of glutamine. Is that it's really important for the immune system and for your gut. It's a fuel that they rely on. And so if you just pull that stuff out, what would you call it? If you pressed it, meaning press is like the ketogenic diet, great, like have at it, keep going. That's not only your treatment. I find this might be a tell, but like a ketogenic diet, it's not only a treatment, it might be a preventive strategy. And you can stay on that. The adverse effects are maybe lower rates of diabetes and other metabolic diseases. With drugs, including like a glutamine agonist that is pulling glutamine out of your system, the argument is that you're going to have to pulse that. The same way that like with chemo and radiation is that you get rounds or courses of chemo or radiation because otherwise they'd probably kill you. You need time to recover from chemo and radiation. Effectively, they're trying, the strategy is they're trying to kill the cancer cells without killing you.
Yeah, there's a lot of collateral damage.
Yeah. And so with the prevention and the treatment, I would say with a ketogenic diet as something that may be beneficial in treatment, and let's just say standard of care, and may be beneficial in prevention, I would say please don't try this at home. Don't start taking chemo and radiation if you think it's going to be a preventative strategy, because those things clearly raise the risk of cancer. That's like well known. So that's one of the problems with a lot of chemos and radiation. They call them secondary cancers. And so while they may kill the existing population, it raises the likelihood that there's going to be a secondary cancer in whoever it was that got that treatment.
And so with the press pulse with the glutamine, it's because it's an essential amino acid. And basically what you're saying is like, you'd have to let your body recover with the glutamine for a period of time and then take it away and continue on in that fashion. And hopefully it acts as a solid therapy.
Yes. Yeah. So that would be the pulsing. But even like hyperbaric oxygen, I think is another one. And I think they'll pulse that in rather than doing hyperbaric oxygen therapy every day. Hyperbaric oxygen is really interesting because you're exposing yourself to very high levels of oxygen, and the way that the proposed mechanism of action for hyperbaric oxygen is under nutritional ketosis, it'll selectively kill cancer cells, and it kills cancer cells through very high levels of reactive oxygen species. And that is the mechanism of action of radiation. And so that would be you'd say, okay, well, if you can selectively do it, that's great. But I still think that there's a pulse component to those things. I think the one, so fasting would be an example that I think Tom would be, periodic fasting, you would say. That's arguable. I don't even say arguably, although there are people that, one guy did it for I think over a year.
Over a year, I think the longest one is over 300.
A long fast. But yeah, presumably that would be something that if fasting was like one of the tools in your bucket, it is something you would have to press. You'd have to put yourself back on some food from time to time. Whereas nutritional ketosis, that's every day. So going back to that mechanism thing, there's this oncogenic paradox on the left-hand side. And so you have all these different risk factors, right? Cancer risk factors. All of these different risk factors. Here we have. This is effectively the definition. A specific process, which is malignant transformation. We talked about those hallmarks of cancer a little bit. These specific hallmarks can be initiated by a plethora of unspecific events. So radiation, asbestos, rare inherited mutations, that's what we're talking about, or we haven't yet, but hereditary cancer syndromes, irritation, inflammation, chemicals. And so that's this paradox that you have this very specific process that seems to be brought about by all these unspecific ways. And this is The Emperor of All Maladies. This is Sid Mukherjee, which is excellent book. The Pulitzer Prize. Won the Pulitzer Prize. It's an excellent biography of cancer, aside from Warburg was left out. Kind of in the Q&A though, because I got a newer book. He has a paperback. It has a Q&A. He does include a Q&A, and whoever does a Q&A, they're like, "What about Warburg?" And he tries to explain it or explains it, and just said,
"Okay, that's good."
Yeah. He said it was like. I think we'll get to this, but almost as if it was like the cancer metabolism field is just like it's burgeoning now, but we couldn't get a final word on it. But he asked the same question basically: what beyond abnormal dysregulated cell division? So like what is cancer? That's what cancer is. It's dysregulated cell division. That's like if you want a pithy definition. So what beyond that is or was the common pathological mechanism underlying cancer. Like, what is the thing that is driving this? And here's Tom, and he says, oh, we solved that. He says that the protracted loss of oxalase following mitochondrial damage is the common pathophysiological mechanism responsible for the oncogenic paradox and the origin of cancer. And so that's impaired respiration, compensatory fermentation. And so now we have these cancer hallmarks. And to the left of it, you can see that there's compensatory fermentation. And so the cell is fermenting. And you have, so what are the hallmarks of cancer? There's 3 papers. I'd say there's 2 papers, more or less. There's the 2000 paper, which is in the middle. That was the original paper by Hanahan and Weinberg. And then in 2011, they have the hallmarks of cancer, the next generation. It's been cited, I don't even know, almost 90,000 times. It's a lot of citations. And then more recently, 2022, Hanahan alone published an update called it New Dimensions. And so this is 2000. We talked about it, that they believe that virtually all cancers must acquire the same 6 hallmark capabilities. And they're on the left. I got them in the next slide. And so these are the ones that you'll see it in the mechanistic slide again, but it's basically self-sufficiency in growth signals. They evade any growth suppressors, limitless replicative potential. So normally you have cells that they have telomeres and they say this and they die. These things, unlimited replication, they can keep dividing. Sustained angiogenesis, so they sort of give themselves a blood supply. Apoptosis evasion, so normally if these cells are all messed up, they're mutated, you'd think, why isn't there programmed cell death? Seems like they have some type of evasion capabilities. And then number 6, which I would argue is like, this is the only one that sort of is a demarcation between like benign neoplasias. Those things have those first 5 hallmarks. Number 6 is what we should really care about, but nevertheless, it's in there, and that's tissue invasion and metastasis. And so 2011 comes along, and they go, oh, look, inflammation, although that's enabling a characteristic. This is when they brought in, to 2011, they called it an emerging hallmark, deregulating cellular energetics, and then also avoiding immune destruction. So it seems like they're able to evade the immune system as well. Like they have these mutations, they're abnormal. Why isn't the immune system picking them up and taking out the garbage? On the right-hand side, they have these enabling characteristics, would sort of be upstream, which is they think it's genome instability mutation, which triggers effectively all those other hallmarks that we talked about. And they also acknowledge that inflammation may be doing something similar. And so this is in their paper. This is the emerging hallmark. And they write, Warburg first observed an anomalous characteristic of cancer, energy metabolism. This was even, so I'd say arguably like in 1920s, but 1930. So this is their emerging hallmark. We're like 1930 is when this guy, it's odd, right? So the existence of this metabolic switch in cancer cells has been substantiated in the ensuing decades. So it's like, yeah, well, you know, in a way, I think like, well, don't trust but verify. Well, we looked at it now, and you know, we agree with Warburg and all the other researchers who subsequently showed it after him. And then I have such reprogramming. We'll get into that too as well, like that word that they're using, the reprogramming. So if you look here, we had those original 6, and now you have 7 through 10, one of which is that energy metabolism, which they're acknowledging that in their own way, reprogramming energy metabolism. We'll get to like the distinction there, because I think it's really important. This was Hanahan in 2022. I'm doing this for posterity, but there's one in here that I have a bias towards, and I think it may be relevant to hereditary cancer syndromes, and it's the one on the top left, and it's called unlocking phenotypic plasticity. It's like, I don't know, just leave it to them to make it like teleological and like, I don't know, strange sounding. So this is, I would call it disrupted differentiation.
So this is unlocking phenotypic plasticity in more simple terms. Okay.
And so I created this is BioRender. I realized like I need to make a slide out of this. So on the right, I'll say, so in the middle, you got a normal cell, like a normal cell differentiation, like what normally happens. Your intestinal cells, for example, you're turning over. It's like, I forget that old thing about like a boat with all the wood planks, and you replace the wood planks like one at a time. And over time, you've got the same boat theoretically, but all the parts are new. The intestine's turning over every week or less. And so how is it turning over? It has a population that they call it the crypt, or the base, and it has stem cells. And the stem cells are proliferating, and then they're differentiating. Sometimes they'll call it terminal differentiation. It's probably a misnomer, but we believe it as they differentiate, and they call it terminal differentiation because now they're like a liver cell. Now they're a neuron. Neuron's a good example of a terminally differentiated cell, actually. So that's the middle. On the right-hand side is that's like the canonical look at a transformed cell. What happens is you have a mature normal cell. That cell gets altered somehow. I put genetic or metabolic on the question mark, or pick your poison. And something happens to that cell, and it becomes a cancer cell. And on the left-hand side, this is also malignant progression, but you're looking at it in reverse, which I find very compelling, actually, in many cases, which is you have this population of stem cells. And these stem cells, in order to become a normal cell, they need to fully differentiate, like the middle one, into a fully differentiated cell. And the argument here is somehow that differentiation is disturbed or blocked. And so you can get a cancer cell that arises from an undifferentiated cell that fails to differentiate. We mostly think of, if not, I think most people think of this, we think of a cancer cell as we have these normal cells, liver cell, pancreatic cell, lung cancer cell, breast cancer cell, and that's a normal cell, it transforms. And we believe, and looking at the data and looking at stem cell metabolism and embryogenesis, I do think that it is likely caused by impaired same thing, mitochondrial respiratory maturation, which is that you'll see even in something called induced pluripotent stem cells, where you can induce an old cell into a stem cell that's undifferentiated and can differentiate into any other cell besides your sex cells, your germ cells. And what happens to those cells? Like, what's the phenotype? Their mitochondria, basically, they become immature. They're like small in number. They're poorly distributed. It looks like abnormal mitochondria, except that it really is like it's immature mitochondria. And if you were able to watch this movie, it does look like these cancer stem cells, normal stem cells, in order to proliferate, when they do, you'll see metabolism in standard textbooks, articles. They rely on, they'll say glycolysis. I suspect they're relying on substrate level phosphorylation from both cases. And then at some point over time, that mitochondrial substrate level phosphorylation is overtaken by oxphos as the mitochondria develop, as the mitochondria mature, and it becomes a fully differentiated cell. And it really is. Tom has one of his chapters, he calls it mitochondria, the ultimate tumor suppressor. I think he's right, is that the mitochondria is effectively what keeps cells in check to not proliferate, which is probably their default state from an evolutionary perspective. That is, mitochondrial oxphos is effectively what keeps things in line. And if you mess with that, that's
really interesting.
Yeah.
And okay. And so the stem cell, like the undifferentiated stem cell, I guess it's not relying heavily on oxphos, or it can't, because it hasn't selected itself for a particular tissue and become part of that tissue. Is that the idea? it can't access the oxygen that's being pushed all around the body quite yet. And so, but it does have a job to do, and it needs to differentiate. And so it's going to upregulate glycolysis to get that energy in order to do so.
What I think in part is that you have, what do they call them? They'll call it like the stem cell niche. So I have this slide here actually looks at it a little bit. So on the bottom right, you have CBCs, which are crypt-based columnar cells, but just think of them as stem cells, right? Is that they have like these little niches, and they say oftentimes they'll say that these cells they're quiescent. So they're just like they're hanging around, and they're not doing a lot of. They don't need to do a lot of they don't need a lot of ATP. It's almost like they're almost like at a standstill, but they're alive and they need a little bit. What I think is, in part, your energy demand, the energy demand of the cell is going to dictate the surrounding mitochondrial population and the capacity of that population. And so if you have these quiescent, dormant quiescent cells, there's sort of like there's no reason to have, that spaghetti network that we showed in the other slides. It's actually like not a hell of a lot's going on. And you'll see the argument sometimes in the papers is they'll go, well, actually, like when they're quiescent and they're not proliferating, they can do oxphos just fine. And I think like, yeah, if your energy demand is super low, even if you have like sparse and poor, I don't want to say quality, but basically poorly efficient mitochondria, you probably can get by with like a little bit of oxphos, and you don't need a lot of machinery for it. But what happens is when they go from the base and they work their way up, in order to proliferate, in order to divide more or less, they basically need to double themselves. They need to double the building blocks, which effectively doubles the energy requirements. And so there's an increase in the demand for energy. And I think that itself, you get the fermentation metabolism, and part of this fermentation metabolic, you want to do mitochondrial biogenesis. You can do a lot of glycolytic workouts that light up the substrate level phosphorylation, things like that. One of the compensatory responses is mitochondrial biogenesis, because you can't, nor do you want to be running on substrate level phosphorylation for your workouts. You can't. We basically can't do it for an extended period of time. And so, like, what does it mean to be fitter? Almost effectively is like you're expanding that mitochondrial capacity. And so with these cells, they got to work their way up from that CBC to a colonocyte on the right-hand side, and they'll call it like a conveyor belt, because it is like a conveyor belt. And so they become this fully differentiated cell. And by the time they're fully differentiated, when you look at their mitochondria, especially, it also depends on where it is, but now it looks like it's not perinuclear. It's actually got more cristae. There's more mitochondria. They look more mature, more developed. They're doing oxphos. The cell itself is doing more oxphos, and SLP is at a minimum. And what happens in the colon is it's called a noikis, which I think it means like without a home or something. But when I think of it, it's like, I look at it as like lemmings on this conveyor belt, and these colonocytes, they like fall off a cliff. They jump off of call it like the basement membrane or whatever. It's basically programmed cell death, but they jump off, and it's basically in your stool. It's like where do those, a lot of it, like where do those, I must have a lot of dead cells if I'm turning over my colon.
Pooping it out. Yeah,
it's in your poop. It's in your poop. So you have this constant process of these differentiated cells that jump off a cliff, and they're being replaced by these stem cells. And so you have this thing where there's these two models in cancer, at least for intestinal. They call it the top down and the bottom up. And top down means the cancer cells are rising from the colonocyte, from the top. And that is creating an undifferentiated like cell. The bottom up is saying, no, the cancer cell of origin is the cancer stem cell. And when you have a cancer stem cell that fails to fully differentiate, I find that both of them can happen, but I see no reason why I find it highly plausible that the bottom-up model can work, and there is evidence for that. And I think I skipped past an important slide in this context. This is how you grade cancer. So these are the quote here. I think they'll probably end up in the image. It's from the National Cancer Institute. And so they'll grade and stage cancer, like your oncologist. They typically grade and stage your cancer. And so how do they grade them? They basically grade your cancer based on how they look under a microscope and how differentiated are they. So the more differentiated they look, the more like let's say we're looking at liver cells. The more they look like liver cells, the better off your prognosis. The other side of the spectrum is grade 4. The closer basically they look to like progenitor cells, they're indistinguishable. You can't even tell that they're a liver cell anymore. And so I find it interesting that you don't like this. The grading is also, if you just think of it as a normal cell that becomes a cancer cell, and that cancer cell over time becomes more and more aggressive, it'll probably represent this sort of stages of grading. But it doesn't preclude that you could have started with an undifferentiated cell that the idea is for it to fully differentiate, but somewhere along the way it didn't fully differentiate. So now you have a cell that's somewhere in this grading pathway that shows up as a cancer cell.
Right.
And I think that's probably relevant for a lot of these hereditary cancer syndromes, but also whether it's colon cancer. Like we can talk about, we only get colon cancer when we're older, let's say. Say it's like 60 years old. Why don't we get it? Why don't more people get it when they're in their 20s and 30s? The somatic mutation theory would say, well, it's just more time. And so that's just more opportunity for mutations to occur and for you to get that dream run, as Davies points out, that it's more time. Like you're playing the lottery more times. That's the argument. In the case of a mitochondrial metabolic component, you'd say, how is it sort of like chronic? How is it chronic? This is what I thought. How is it chronic oxphos insufficiency in a cell that like it's 5 days old and it's gone? You could argue that like something happens where it evades apoptosis and everything like that. But the other component is that there's a Lamarckian component to this, that in some cases you can have the cells, the differentiated cells, that they can de-differentiate to become a stem cell, or the stem cell population itself. Their mitochondria, if their mitochondria like has wear and tear over time, is that as those cells, you can say as those undifferentiated brand spanking new cells divide into a differentiated cell, they're carrying mitochondria that actually may have the slings and arrows of its past, which I find kind of remarkable, because Lamarck is like synonymous with quackery, because you think just because I lift a bunch of weights, I got big muscles, so my kids can have big muscles. That's how people think of Lamarckian evolution. But you think just because I have damaged mitochondria, some of my ancestor cells might inherit it. Yeah, that's a possibility.
Okay, so we're rounding out the second period here, Bob.
Okay. Let's see where we are. Okay, not bad. All right. So now we're past the oncogenic paradox. And here you've got reactive oxygen species. And hopefully I can get to some caveats here. But the way that I said that the way Tom throws around Ross is he's a cancer researcher. And he says Ross are carcinogenic and mutagenic. I don't think anybody disputes that. And that would cause significant damage. Basically, lipids, proteins, nucleic acids. Effectively, you can damage DNA with that. No question. Nobody argues with that. So the nuclear genomic instability would arise as a consequence of ROS damage. And so how can you get, so in part, he would probably say, how can you get a cancer cell with no mutations? Those are very rare. But how come you get cancer cells with millions, like literally millions of mutations? That's just like the dream run, or that's because DNA repair mechanisms are out of control. It can be in part because it's a feed forward vicious cycle. And if you do have cancer cells that blow out a lot of ROS. And we'll get to this too as well, that it's almost this idea as well that we know, Pete. I won't confuse matters, but I'll say a little bit of like high levels of ROS, but not at the levels Tom's talking about. That actually promotes like proliferation, tumorigenesis. You go higher than that, and they call this a paradox. They say it's almost like a ROS paradox in cancer. When you go higher than that, it causes cell death. And that's actually what we do for treatment, like radiation, chemotherapy. We're actually trying to create a lot of ROS. So are we trying to create cancer? No, but we actually do in the bargain. That's secondary cancers, things like that.
Right.
But you can have elevated levels of ROS. It depends on that triangle or the degree. Elevated levels of ROS that can promote proliferation. And then as they get higher, that type of signal is basically like cell death. It's just like, not just like, but I talked about cyanide, cytochrome C. Part of that process is that thing takes off, it's untethered, and you get cell death that way. No ATP. So here, this is the oxphos. So this is retrograde signaling. So retrograde signaling, I mentioned it. That is the mitochondria signaling something to the nucleus. So what does it signal to the nucleus? Like, I'm not creating like ATP crisis, not enough ATP, or too much ROS. So either way, like I've got to downregulate my oxphos machinery, because when I'm doing that, I'm making ATP, I'm blowing out a ton of ROS, and or there needs to be some type of compensation. And that's when you see something like HIF. That's probably, I think, hypoxia inducible factor. But if HIF goes up, HIF is constantly, actually, it's interesting. It's constantly expressed. So it's we expend a bunch of energy making HIF, putting it out, and then like putting it out into the ether, let's say, and then we squash it immediately if there's enough oxygen available, call it. And so why is that? Why do we do that? Because in part, if oxygen isn't available, if there is a crisis, like we're going to need HIF, because that is where you're going to get fermentation, all these fermentation pathways, in addition to like angiogenesis and all these other things. So the idea here is if you have this persistent fermentation metabolism, you're actually going to upregulate these pathways that put the cell into you could call it a primitive state or the default state, which is a proliferative state. And in part, Davies and Lineweaver, they sort of point that out, like from an evolutionary perspective, default state of cells look a lot like cancer cells, just bacterial cells, like divide and reproduce. That's all they do. And so in this case, you've got not only the fermentation, glucose and glutaminolysis. So like CMYC is a great example of, it's called an oncogene, and it's something that promotes, like it lights up and helps activate and create glycolysis enzymes. It does the same thing with glutaminolysis. And so it gives you these pathways to create ATP in a crisis. And so it probably, in part, you compensate from exercise, you do the same thing. That's an acute thing. In this chronic basis, you're effectively switching this on long term. And so now you have oncogenes, therefore, become facilitators of increased cytosolic and mitochondrial SLP that drive dysregulated growth in cells with insufficient oxphos. And so the oncogenes, the tumor suppressor, like these, even if they're not mutated, that they're just activated. Like CMYC is a great example of, often, I think it's more often than not, this seems like abnormally activated, but it's not mutated in cancers. And so it's something that there's reprogramming cellular metabolism is what Hanahan and Weinberg say, and they probably attribute it epigenetic means, that type of thing. And on the bottom here, this is where you're looking at the minus 56 kilojoules. Just think of that as like sort of like that's what the cell had, like that's 56 ATP. That's what the cell basically needs at virtually all times in order for sustainability. And what happens here is it looks like progressive decline, that's the green, the oxphos, progressive decline in oxphos capacity. And so less ATP is coming from oxphos. And when that's happening, if the cell doesn't die, the reason why in part is because you're getting a compensatory ATP supply. And that uppercase T, it's like, it would be nice to know this is the the line of demarcation,
like the tumor
Rubicon.
line. Yeah.
You cross it. This is where you cross the Rubicon. But they just, he puts it like it's an arbitrary line, but it's a threshold where the shift from oxphos to SLP would become irreversible. Although I hold out that they're now like mitochondrial. We talk about those cytoplasm, those experiments, right? You stick a healthy cytoplasm. There are now these mitochondrial transfer experiments. And so I am curious, like, imagine if you can transfer these healthy mitochondrial cytoplasmic populations into your cells. It's possible. Who knows? Like
SLP, for those of you guys watching, it's substrate level phosphorylation. So that's the glycolysis and glutaminolysis.
Yeah, and the phosphorylation. So OXPHOS is short for oxidative phosphorylation.
Yeah, exactly.
That's basically one of the P's to make ATP, which is the T is for tri. You're getting 3 phosphates. Typically you have 1 or 2 and you stick one on there. And so substrate level phosphorylation means you're using a substrate instead of the electron transport chain to make that ATP.
Bob, keep going. I'll be right back.
Okay. Go to that last one. Up. So, and this is the quote here. This scenario links major cancer cell hallmarks to an extra chromosomal and epigenetic respiratory dysfunction, thus solving the oncogenic paradox. And that's what he was saying at the beginning or at the top of this, which is damage to respiration, ultimately damage respiration, compensatory fermentation, and that the fermentation phenotype or metabolism activates or sustains a lot of those cancer hallmarks that you see on the right-hand side. Okay, so now we're actually into hereditary cancers. This is looking at the percentage of overall, what's the percentage overall of cancers where we compare so-called sporadic cancers, which is most cancers, so not familial, not hereditary. And then you have familial cancers, and then you have hereditary cancers. We're looking at hereditary cancers, which makes up a fairly small sliver of all the deaths, 5%. Sorry, of all the cases. So here we have what proportion of cancers are caused by the environment. And here I have highlighted the germline prevalence. These estimates, that's the 5% that you just saw in the previous slide. What's interesting too is you'll notice, for example, like lung cancer. We have lung cancer is clearly, that's the leader in the clubhouse as far as cancer deaths. Not necessarily cancer cases, but cancer deaths. But from a germline perspective, from these hereditary cancer syndromes, there aren't any that light up as far as lung cancer. Colorectal cancer, there are more. But for lung cancer, you can see that the germline prevalence may be as low as 0.3%, accounting for like less than 1% of all cancers, which is kind of remarkable when you think about in the context of lung cancer death. And then you have the familial prevalence, which is first degree relatives, things like that, where they haven't necessarily identified a genetic component, but it clearly seems to run in families, and there seems to be a higher risk within the families. I do think they're probably doing their job and their due diligence, but oftentimes the gene environment interaction, I think, gets a little bit fuzzy, because you can say these things run in families, and then you look at the families and they not only share the same genetics, they share a lot of the same environment or lifestyle. And so if there are lifestyle or environmental components, sometimes those things get baked in. So what they often try to do is, some like cool cases is they do twin studies, and it's not the heritability necessarily of twins. They find twins that they're like separated at birth. Right. And so they have the exact same genetics. And now you could say, well, they're not in the same family, and these families look a lot different. And so they try to tease those out. There's not a ton of those studies, but It's 5%. So here's environmental. Age is, to me, it's like age is it a risk factor or is it a number? Is it a number that, so there's a whole thing about biological age and chronological age. In parentheticals, it says over the age of 65, which sort of implies it's chronological age, but that's a little bit tricky. I would keep that out of there for now. I wouldn't say, and talk about a non-modifiable risk factor. Although the biological age people will tell you it is modifiable. You can change your biological age while the clock is chronologically ticking. But here you can see these are, I would say they're pretty conservative estimates, but the percentage of cancer deaths in the US, it's like a third of all deaths are smoking, and this is from 2022. Obesity, 7%. That number seems to be, it depends on when they're how they're determining the risk. That seems to be rising. And then you have next is, and it's not necessarily the lowest, but that's where we have the hereditary cancer syndrome for germline mutations or heritable cancers. But I'll just go to this slide, which is, this is effectively, at least this is a more aggressive argument about what, without even thinking of cancer as whether you think it can be a genetic disease or not, what proportion of cancers are environmentally induced and what are genetically induced. And so genes make up 5 to 10% of it. That's the germline prevalence. That's the hereditary cancer syndromes. That's the 5%. And then you have the familial cancers, which you don't really know, but it seems like it travels in those families. That's the 10%. And so that's the 5 to 10%. And then the other ones, it seems like they can at least be partially explained by environmental factors. And so diet makes up 35% of it. I think the more recent ones do physical activity. It's if you want to talk about loaded terms, right? Well, I would say diet, is number one. And then exercise is probably a number two as well. But I would say diet is one of those most squishy ones. And then I'll skip past these things, but this is showing. So this is here too. This is a slide that I got. It says cancer deaths linked to diet as reported by Willett. I'm like, oh, Willett. That's Walter Willett at the Harvard School of Public Health. So he's probably looking at Nurses' Health Study, things like that. And I know part of that is I'm pretty sure he probably believes that colorectal cancer, so 70% of colorectal cancers are attributed to diet. And I would say probably a non-trivial proportion of that estimate is based off of red meat and processed meat consumption. And I would say I've looked at that data, and that data is bullshit. It's not. I would say in part it's not even wrong. I'd make the argument that the relative risk and the risk ratios, that they're actually making an argument that it can't be a meaningful component. But I just want to, so that previous one, just to give you an idea, I thought that this paper was kind of cool because breaking it down into infections. I'll go back one because I want to do that. This is another one of my projects that I've been picking up on, which is I've been collecting all the quote unquote infection associated cancers. So you have tumor associated viruses and you have bacterial associated cancers. So H. pylori is a good example of a bacterial one. And then for the viral ones, what's the one? There's the H. pylori. So HPV is an example. Actually, all these are examples. HIV is an example. So what I've been looking at is what is the argument for these having a viral oncogenic component to what do these things do to metabolism? Right. Sort of it's like a broken record. And I feel like I'm acknowledging, I am cherry picking. I'm just looking for data that will look for, is there a metabolic component to, it's a good example, that H. pylori, the HCV, what's the HCV? Oh, so hepatitis B, hepatitis C, great examples of things that are those viruses infect you, and it looks like they do things to your metabolism. They do things to the mitochondria. A lot of these things do. So it does look like these things can be, you could call it working, the raise cancer risk through metabolism and not necessarily through, you could call it genetics, that they're sort of inserting their own genetics into you. But I want to look at all of the risk factors. So this is a chunk that's like in the world, the estimates are like, say, 15%, 17% of all cancers are attributed to this, infections and cancer.
I mean, at the very least, it should raise questions for people, right? And then at the most, it then, depending on what ends up being found, the genetics become secondary factors rather than primary.
Yes. And with cancer, they say, I think people just like assume that smokers, if you smoke a lot, you're going to have, your cells are going to undergo a lot more mutations. And I think that's like directionally true. But there are studies, I think it's in, what do they call them? Former smokers. So people who have quit, they look at former smokers, and then they look at incidents of those. So they didn't get cancer, but they were smokers. Then they look at this cohort of former smokers. Some of them were getting cancers, the other ones were not getting cancer, and there didn't seem to be a difference in their mutational profile as far as like driver mutations, tumors, those other things. So I've looked at smoking. There's like a lot of literature. It really depends on like the researchers, because the hereditary cancer syndromes are actually, they might be the most difficult because they're literally defined by the gene mutation, and they just sort of say like, that's a wrap, it's a genetic disease. With something like smoking, you have these people looking at it, and they're looking at the mitochondria, they're looking at the morphology, the how much ATP, like respiration, all those different things. Smoking clearly impairs mitochondrial respiration, which probably doesn't, like, you can almost see it, in people who smoke. But anyway, be that as may, be interesting to go through those things. I haven't yet, so I'm not going to do that. But I want to go to this one: fruits, vegetables, spices, condiments, and cereals with potential to prevent cancer. And I was like, what do they have here? They have like 30, almost like 30, 60, 90. They probably have like 100 things here that are associated with a reduced cancer risk, right? And you think like, hey, that's great. I would say like, hey, this is epidemiology. Like, this is the steaming pile of dog shit that you're looking at. And this is Johnny Anides, who's like, he's one of the good ones, is what I call him, because I think he's an epidemiologist. He's sort of like skeptical, kind of a skeptical epidemiologist.
This is really
which
fun.
I think all epidemiologists should be. I would be really skeptical if I was an epidemiologist, to go like, is what we're seeing really, really there? And so he did a systematic cookbook review. Sounds like almost like a joke, but he literally did this.
Sounds like a joke.
Systematic review. So a systematic review, that's your top of the hierarchy of evidence, whatever. But systematic review is really like you're systematically reviewing normally, say, randomized controlled trials, and you have inclusion criteria and exclusion criteria. And they should be systematic, and they should be sort of homogenous in some ways. I would argue that like there are many limitations to that. Nevertheless, that he's doing this, he's systematically reviewing a cookbook. I think it was a Boston cookbook, and he's selecting at random. I'm going to select at random from this cookbook, like 50 ingredients. And so we selected 50 common ingredients from random recipes. And the results are 80% of the ingredients that they selected, so 40 out of 50, there were articles, right, reporting on the cancer risk. And 72% of them concluded either there was an increased risk or decreased risk. And it was about half, and you can see that the number's far larger than the number of ingredients. So this thing causes cancer, this thing cures cancer, the same thing. And so then he looked at meta-analyses, and he's sort of like he is a meta-analyses person. They presented more conservative results. So when they looked at meta-analyses of these ingredients, so when they like pool the studies together, if you're looking at like cinnamon, you're doing a meta-analysis, meaning you're analyzing all those studies and trying to lump those all in together and then pool them and see what is the risk ratio that you get. And in this case, it it was 26 reported an increase or a decrease. So it went from, what was it, 72% to 26%. And the median relative risks were 2.2 and 0.52 respectively. And so the conclusion is basically many single studies highlight implausibly large effects, even though evidence is weak. Effect sizes shrink in meta-analyses. And so it's basically like, you could say the epidemiologists are fooling themselves if they're publishing these things and they really think that, like, they think maybe eating like 6 nuts every day will extend your lifespan or reduce your mortality risk. Maybe that is the case, maybe. But this sort of suggests against it. But even though, if you look at this is like nutritional epidemiology, almost like in a nutshell. Like when you're looking at your study, you're almost assuredly fooling yourself. But I'd say, like, are they fooling themselves or are they fooling me? Because they're really trying hard to, and I'm biased too. Like I don't know how I would be on nuts, but let's say it was like red meat and I was the researcher. You could say maybe I'm biased and I would be really rigorous if I saw like this looked like it was an increased association or something like that. But if it was if there was no association, maybe I'd just be like, yeah, that's great. Like, check it off in the W column. And I think that kind of happens. That I do think that. So one thing that I would wish for, and I think David Ludwig talked about this. I don't even know if he was in favor or not about this. Your disclosures, your conflict of interest, it would be really useful. And like, I do think, like, volitionally, that the researchers should say, if they're nutritional epidemiologists, they should say what diet they think is best and what diet it is that they eat themselves. Just the same way that they would say, we got funded by the nut council or the dairy industry or the meat industry.
Yeah. Because
people will go like, oh, maybe they're overcompensating the other way, but they should disclose it. Because I think that might be, it tells you the intellectual.
That's a good point. Yeah, that's a good point.
Yeah, I think it's just as real as any financial conflict of interest. So this is where we get to this reprogrammed cellular metabolism. So one of the arguments is one of the reviewers, the anonymous reviewers for this paper, it was effectively like, I don't know.
For your paper.
Yes. I'll say in general, there were two anonymous reviewers. This is peer review. I know sometimes that's getting knocked. I would say that they were, you bring I think most people might be like this, but they were highly skeptical. And I would say, like, equally helpful. Because they were so skeptical, I think it made it really constructive if we just took it as like, yeah, they're going to have problems. Like, they're going to have problems with this being presented when it probably runs counter to a lot of the things that they either believe or they believe that what we're saying is nonsense, except for they reviewed it and they can go look at the references and citations. So one of the things was, one of the reviewers kind of said, like, yeah, no shit. Like, cancer cell metabolism, we know it's altered. Like, we know the Warburg effect occurs. So, like, the fact that you guys are saying there's impaired oxphos and there's compensatory respiration, it's like, we already know that. And so it was like a little bit of a back and forth, but I don't think we made the distinction strong enough. And that was one of the better critiques in a way. Was like, we didn't convey the difference between how reprogrammed cellular metabolism looks like from the SMT perspective, because you can say that they have adopted or incorporated to some extent that energy metabolism plays a big role in cancer. They do acknowledge that. They just say that the upstream component to it is mutations.
Are the mutations,
including
right?
Metabolism. Yeah. So what they say is the mutations. That's an enabling characteristic. The genome instability, immutability. That's what's enabling a rewiring. Like, these are their words, a rewiring of cellular metabolism. Mutations enable the cell to reprogram metabolism. one of the several hallmarks cancer caused by mutations. So they think that the metabolism comes after the cancer, effectively. Like the cell has acquired the mutations, it's now a cancer cell, and now it does its rewiring because it chooses to, which is like that's teleological, and you'll see that, and that you should look for that. That like it'll say like the cell prefers to ferment. Cell prefers to ferment glucose instead of oxalase, even though it's inefficient, because it wants to make building blocks, et cetera, et cetera, et cetera. You think like the cell doesn't want to do anything. Like the cell just responds to its environment. Although I guess they would argue that like that cell is all messed up because of the mutations that it's right. Some of those hallmarks, but
I guess they would argue that the mutation has chosen.
They give it. They just I don't know if it does make it simpler, but they do really. They give everything like a teleological term. Remember I said, all right, I didn't really like the term. It said unlocking phenotypic plasticity, right? It says unlocking, right? Like the cancer cell unlocks, like it. It's not. I don't.
Why was it? Yeah. Why was it locked away in the first place?
Yeah, I just think like when you talk about the fermentation, it's like when I'm at the last grade of a VO2 max test, right? My cells are like they prefer to ferment glucose. It's like they're in a crisis, and what is it's the metabolic milieu that like dictates it, and that's like why they're blowing out lactate. Why would they blow out lactate? Because they're not, the hydrogen ions and the acidity, they're not going to be able to sustain that. That's odd. That's what they say about cancer. They don't say that about exercise. That's why I do go into the exercise physiology field, because you'll pull a lot of the same proteins there's one of the sort of tricky ways that I was at least able to find, if there wasn't a lot of direct evidence, is you could find BRCA1 in skeletal muscle, not just memory tissue. And they're looking at it from the perspective of muscle performance. But then you get into mitochondria, you also get into fermentation pathways. So that's basically a different. So with the MMT, oxalase insufficiency, that's the enabling or essential characteristic. That's what causes SLP dependency. And that is, I think, it's straightforward. You could call it elegant. The issue is there is a large field, not even a large field. It's a small field of a lot of large people, let's say, that like a lot of them I happen to like too, but they're cancer metabolism, like not every aspect of the research, but they say the mitochondria is fine. And it is this idea, like the mitochondria in these cancer cells, they're fine. They're just effectively, it's like they're opting for substrate level phosphorylation, because substrate level phosphorylation, sort of to Tom's argument, is like when the cancer cell does that, it actually does. It gives them all these building blocks. It gives them the speed of ATP respiration in order to proliferate all that stuff. It's almost like you are in a way making the same argument. And so that anonymous reviewer, in a way, he's like kind of saying like, aren't we like simpatico? And I said, no. I said, not really. And I said that it's better if it's the more you can separate these things in order to compare them, rather than to just like Frankenstein the thing, which is, I do worry about that. And I think that's what a lot of people are saying. They're like, well, it's a metabolic disease, it's a genetic disease, it's an immune disease. So let's use all those buckets to fight cancer. And it really, I agree with Tom, where it's really like, you really want to understand like the etiology of the disease. Don't just start, going after what might be effects.
Yeah.
Because those effects might get larger and larger and larger.
Yeah. Yeah.
Okay. So this is like the central graphic. This is, we've effectively, it's going to be applying all that stuff to hereditary cancer syndromes. So I've just broken it up because the figure is unwieldy. This is the first part of the figure. So we're looking at, this is a cancer predisposition gene. This is the alphabet soup of just think of BRCA1 right now, or P53, are two popular ones. That's where you'd stick in that cancer predisposition gene. This example is, we say non-penetrant. In this case, non-penetrant means it doesn't eventually or effectively cause cancer. And so what happens here, you have a cancer predisposition gene. There can be OXPHOS impairment, but it's not sufficient for malignant transformation. So there are cells to varying call it fitness in terms of their mitochondrial capacity. And so as long as the mitochondria, you looked at the arbitrary T number, is maintaining, say, sufficient OXPHOS, cell growth and regulation is maintained. Then on like the 2 here, is that the downstream somatic mutations without malignancy, is that you may see more mutations in these particular cells. So somatic mutations on top of the predisposition ones. This is an example of, I guess we haven't found it, but this is a cancer predisposition gene, or the one that ultimately goes on to cause cancer. This is the cancer predisposition gene. Ultimately, like, that thing puts it over the edge in terms of its OXPHOS efficiency. And so now you have, if you look at the SLP on the bottom, compensatory SLP and the OXPHOS insufficiency. OXPHOS insufficiency precedes it, SLP, and then you get dysregulated growth and malignant transformation. Then you have the CPG and additional cancer risk factors. I would say those two are like hypothetical. And so, of course, just living in the world, there are other factors, there are other components in the environment that may be competing, and at some level, they could be impacting risk. So on the left-hand side, we basically saw that in figure A, or the sub A. That's where we looked at, okay, we got a cancer predisposition gene. It might be impairing OXPHOS, but not enough to result in malignant transformation. But now let's say let's say you're BRCA1 and you're a smoker. And so that's cancer risk factors, and that's the oncogenic paradox. You have that multitude of risk factors. But let's say like you smoke on top of having a BRCA1 mutation. That additional risk factor on top of mitochondria that's, say, not operating at 100%, it's already knocked down in terms of its efficiency. So you have, in this case, now you've crossed that arbitrary threshold, and now you get a downstream somatic mutation. This is like Mary Claire King saying, like, how can it be, if the data is, like, we're not making a mistake on the data, how can it be that we have remarkably lower rates of breast cancer in BRCA1 carriers prior to 1940 compared to like 1991? And they looked at smoking, and they looked at like alcohol, and there was a lot more of it in the newer cohort, let's say, which makes sense. Like if you think of like the smoking, cigarette consumption rates. And so that's what basically this is saying, is that yes, like having a BRCA1 mutation, certainly at least in the United States, maybe not if you're a chimpanzee, and potentially not if you're a hunter-gatherer. I don't know, like maybe you'll never see like you won't see anybody that sees the day, even though they have a BRCA1 mutation, that'll be enough to push them over the edge. However, when you live in a population that you have a BRCA1, you are predisposed, especially, I would say, and the argument we make is BRCA1 clearly, like normal BRCA1 clearly has to do with mitochondrial integrity, mitochondrial homeostasis, OXPHOS, all that stuff. And so if that's impaired, or you lose that function, and you have impaired mitochondrial OXPHOS, and then you have other cancer risk factors that are also putting pressure on that same thing, where it's impairing OXPHOS. That is it's called the gene environment interaction. And so those two things, you could say they play with each other, but I'd say like it's in effect, the gene itself is expressing a phenotype, which means impaired respiration. And so you have two of these things Even that was the Mary Claire King. That was if you pull, at least from an epidemiological perspective, we can't do randomized controlled trials at this point. Have to a placebo, have to like a two carton of Marlboros a week. But from the observational perspective, they go like, if you layer on these things, or if you find populations that smoke, lower socioeconomic status, et cetera, who are BRCA1 carriers, lo and behold, your so-called penetrance, which is lifetime cancer risk, it could be very different in a different population, and that is environmental. It is non-genetic, and that is like, I don't think it's not even controversial. And so the fact that these two things can play with each other, I think is really important. So when we're talking about, like we can grant it that you're at an increased risk for breast cancer with BRCA1, or Lee-Fraumeni syndrome, and there's multiple cancers with P53. But the more that you can lower cancer risk factors, arguably, we argue, that affect oxphos, the lower your risk. This is cancer risk factors. So this one, acquired cancer risk factors that do not result in cancer. So a great example, and I think it changed. It depends on how much you smoke, but we saw it earlier too. Cigarette smoking is accounts for, it's about 85% of all lung cancer deaths. Almost 9 in 10, right? Cigarette smoking accounts for almost 9 in 10 lung cancer deaths, but only 1 in 10 smokers get lung cancer. So that's an example of you've got a cancer risk factor that I think a lot of people would argue. I'm not going to argue with them necessarily, that smoking is about, like, you want to get cancer, like, light them up.
Yeah.
If you do that, I mean, a 10% risk of a lifetime risk of lung cancer. Although again, there are studies that they'll stratify. They'll look at dose response. And if you do smoke like 2-3 packs a day, I don't know how anybody does that. I can only do a pack a day, like back in my high school days. So it's like a full-time job, just like lighting one off the other. Be that as it may, this is, again, in effect, you can smoke and your mitochondria probably, they might not look pristine, but it may be, you do have, it's the alveolar. Now you're looking at lung cells, and interestingly enough, most of cancer cell transformation are like they're epithelial cells, and they don't have like the same dynamics and range of, like, say, a muscle cell. And muscle cells are thought to be terminally differentiated. I don't think that's not necessarily true, but there is heart cancer, there is muscle cancer, there's rhabdomyosarcoma. They're exceedingly rare compared to lung cancer, colon cancer, breast cancer, and those are more epithelial cancers. And those typically have like relatively low oxphos requirements to begin with. But if those things get knocked down, I don't think they have a lot of wiggle room in order to compensate just with oxphos.
Bob, we got 15 more minutes. And if we have to continue, if you got a good chunk at the end, we can always do a second round. But if you're close to the end, then we can push through for 15 and see if we can wrap it up.
Okay. So this is
up to you.
Yeah, I'll do another 15 minutes. At least get through this figure. Mm-hmm. So this is cancer risk factors that result in cancer. This is similar to the slide where we showed, okay, maybe we have a cancer predisposition gene, and we layer on a cancer risk factor, and so maybe the additional risk factor knocks them over the edge. In this case, you've got somebody that doesn't have a germline mutation. In this case, okay, let's say like you're a smoker and you've got this cancer risk factor, and it's impacting your mitochondrial respiration or integrity. But it's not enough to knock you over the edge. And then you have additional cancer risk factors. Let's say it's seed oils, Pete, or sugar consumption. But I would say whatever it is that ultimately inflates the fat cell, and you get ectopic fat, and you got ectopic fat everywhere, and I think you're a metabolic mess. That'll probably impact mitochondria. So you have that layered onto smoking. You're probably multiplying your risk. And so in this case, it took a couple of, you could call it like a couple of hits of two really substantial risk factors. But again, in this case, it's not that these risk factors are causing mutations. In fact, it's like most of the common carcinogens that we find, they're not mutagenic. And so it's a hard thing for them to reconcile that if these carcinogens are causing cancer through from the SMT strict perspective, they should be causing them through mutations. It doesn't seem like that's happening. So this is just a quick summary. We didn't get into all these things, but it is interesting to look at. These are the lifetime cancer risks on the right-hand side. The ataxia telangiectasia, I never say that one. That one's that's in the supplementary. So it's a lifetime risk of 10 to 13. So I'll highlight the bigger ones. The second one is in the supplementary, but it's supposedly like 8 out of 10 people with this mutation. Bloom syndrome, again, it's variable, 8 out of 10. These other ones. So it's another one. Here we go. So just wanted to highlight quickly FAP, familial adenomatous polyposis. That's one that a lot of some estimates will say it's fully penetrant, meaning like if you have this mutation, you get it. The literature just doesn't support that. And in fact, you actually have two kinds of FAP in the literature, which is you've got FAP and you have AFAP. And AFAP is called attenuated familial adenomatous polyposis. And attenuated FAP is defined by the number of polyps that they find in your colon. And so when I saw that, I realized so this isn't fully penetrant, because even though they're basically selecting, you have to have over 100 polyps in order to be diagnosed with FAP. And so here we've got all these things. We can blow past the snapshots. I'll leave everyone with who is it, John Nash? John Nash and his drawings in his garage. This is my John Nash drawing. I try to just map everything out, but ultimately you look at. I try to put in squares, and I recently updated this because I realized I didn't have the supplement one on the bottom. So I'm going to move back. But if you were to look at what is the hallmark, because I would say Tom's stuff made sense. I just didn't know if it made sense from the perspective of hereditary cancer syndromes and what is going on with the alphabet soup of these hereditary cancer syndromes. And it does seem like, lo and behold, they either downstream here, they impair OXPHOS, or they promote substrate level phosphorylation, or they actually, one of these so-called oncogenes, they promote the activation of, say, MYC and ROS and mTOR, those are the ones in the circle, or HIF1. So they promote, either way, they promote a fermentative phenotype, but they all seem to converge on this metabolism here. And so I found that, I found it pretty striking.
Yeah, that's very striking. Either they're decreasing mitochondrial oxidative phosphorylation, decreasing beta oxidation, increasing glutaminolysis, glutamine uptake, glycolysis, or glucose uptake. That's for sure striking.
Yeah, I did. And this wasn't comprehensive, but you can see too, on the top right, you see the arrows to HIF. That to me is
hypoxia inducing factor.
Yes, that's a tell, but I'll go to one of the things here too. So this is the role of ROS in cancer and the RTG, is that it's increasingly recognized that the retrograde response can be a few different factors, but the retrograde response, I think we're learning this as well, is it's mitochondrial ROS, not the carcinogenic mutagenic, but it is the signal. And so one of those signals, what keeps coming up is HIF1. And oftentimes they'll talk about it in the context of hypoxia, which makes sense because in the presence of oxygen, that's basically the substrate that you need to knock down HIF, that that more or less is like, if I have plenty of oxygen around, I don't need HIF. If I don't have a lot of oxygen around, HIF is stabilized. And so you have this fermentation metabolism. However, like the thing about this is, and this is Nav Chandel. Nav Chandel, like in, I think like 2000, he's a cancer metabolism research. He was saying that HIF is not only activated by a lack of oxygen. Reactive oxygen species do it. This sounds like like a Warburg thing, right? Like even in the presence of oxygen, right? And so increased ROS will increase HIF even in the presence of oxygen. And then you've got that ROS. So what else might do that? I started looking at that. So succinate, right? Succinate and fumarate. These are TCA cycle metabolites. That if these things accumulate, almost like ROS, they're signals that something is messed up with it. I would argue something is going on with metabolism, and it's effectively like it's a call to the nucleus. So if you have like succinate, and this is also in the presence of oxygen. So a couple of the PPGL is one, HLRCC. So PPGL is a hereditary cancer syndrome, and that's one of those things that came on relatively new to the scene. That is a mutation in the TCA cycle. That is succinate dehydrogenase. That's complex 2. And so that was like, oh, maybe the mitochondrial metabolism is really important. And then the other one was fumarate hydratase, this HLRCC. And so those two things, you blow out a bunch of succinate and fumarate, probably in both cases. That is like a retrograde response. It signals just like partly with ROS as well. So you have both those things. But it signals HIF, it signals fermentation metabolism, in part because I think succinate, fumarate, whether you're talking ROS, or even you're talking like that calcium accumulation or ATP deficiency, all of those are in effect, there is some insufficiency in the mitochondria, and it is signaling to the nucleus, like, we got to shift things over here. And that's ultimately what happens. This is what the idea of this retrograde response is. So I wanted to double click on that. And then also just for the perspective of ROS is that you've got like the left-hand side, supposedly homeostasis. So you can have it depends on like how you're looking at it, right? So you can have high basal levels, low basal levels, and those can oscillate. With tumorigenesis and proliferation and growth, you have higher levels. I suspect that like it's almost like you're in the redox state. Like when you think about like, well, that's enough ROS to inhibit, let's say, insulin signaling. Well, imagine like the cell unavoidably is like creating enough ROS that is effectively like a growth signal. And this is how, from an evolutionary perspective, not controversial, a low level of ROS, effectively, you'll see proliferation of bacteria, you'll see proliferation of cells, and then a very high level of ROS. I might have a slide for this. Bacteria? No, maybe not. Okay. But a high level, if it's too high, there's cell death. But if there is ROS initiated, and then those experiments, they'll add catalase. It's an antioxidant. And so they'll give it they'll give the bacteria, like it should be a feeding frenzy, tons of nutrients, right? And they give them an antioxidant, and the bacteria doesn't eat. You take away the antioxidant, the bacteria eats. And so it's more elegant than I just said, but it's a compelling case that the ROS are effectively, it's a signal that there are nutrients around. We can consume these things, and we can grow and divide because there's plenty of nutrients around.
There's an abundance of it, yeah.
Yeah. I think, let's say like we're at 3, we can stop there. Unless we just go to.
Do you have important slides that you want to share with everyone on another one, or do you have a summary?
I mean, we could wrap it up.
Here we go. Conclusions. Yeah.
Yeah, we could probably talk about it at another time, because I think from the exercise perspective, we can go through these and talk about how these tumor suppressor genes are like involved in exercise metabolism. You could
I
call
love
them.
that. So let's do that at the
Yeah.
you can conclude right now, and then we'll pick up on that topic for the end of February.
Okay.
So if you
So the conclusions is we're just basically rehashing the different things, but we're suggesting that oxphos insufficiency is a necessary condition for inherited pathogenic gene variants to increase the risk of dysregulated cell growth. If an inherited variant does not compromise oxphos, it wouldn't elevate the risk and it would not be recognized as a cancer syndrome. So part of it is like, if this theory is correct, a part of me was like, how come I feel like I'm, in a way, like I feel like I'm cherry picking. I am looking for metabolism, but those papers could have said oxphos not impaired, no aerobic glycolysis / Warburg effect going on. Like it just keeps lighting up. And I can imagine Tom, like if I was doing the paper with him at the time, rather than just collecting it all, he'd say like, yeah, because that's what this is. That's what you're going to see if these things do raise the cancer risk. So that is the upshot. I thought there was just one more line that I think is an important takeaway. This. Okay. Because I think that I don't know if it was one of Tom's papers or something, but thinking about this, like, is this thing necessary? Is this a necessary and sufficient cause? And so if cancer was if cigarettes were a necessary cause of lung cancer, then everybody who has lung cancer must have smoked, right? That's a necessary. If it's sufficient, it's anybody who's ever smoked gets lung cancer. And so in this case, I'm looking at insufficient oxphos and I'm thinking, I'm guessing at least, I think it's the case, you can have insufficient oxphos without malignant transformation. And potentially, like, over time, your oxphos is partially impaired, let's say. And maybe there's an arbitrary threshold, but if we're playing by the rules of the game, insufficient oxphos, it wouldn't be sufficient, but it might be necessary, right? And so if it's necessary, then maintaining normal oxphos is a necessary and sufficient way for preventing cancer, which means, like, if oxphos is not impaired, then you can't get cancer. That can be, like, logically true, even though you can have impaired oxphos and not get cancer, meaning, like, it's not a necessary cause and not a sufficient cause, which I think is powerful if you believe that these cancer risk factors and these cancer genes, these might put you behind the 8 ball. There's still something you can do about it, and certainly for the general population as well. So, like, the best way to avoid a fight, don't be there. Like, that is, when I look at all, like, when I go nuts about the American Cancer Society, I was like, just you can't move the needle on it on any of this stuff, apparently, except probably cigarette smoking. I'm like, just put all your eggs in that basket. It's still like 1/3 of all lung cancer deaths, and we think that, like, that's 1/3 of all cancers. And if smoking is causing 80% of them, put all your efforts into that. Just, like, whatever. Like, I'm not a draconian socialist, whatever. I'm like, just pull them out of the environment and say, you want to smoke? Go to Canada.
Yep.
Just kidding.
Yeah. Go to something about the cold air.
Yeah.
It might actually help. So This is good, Bob. This is amazing. The reason I love this paper so much is because, I mean, if whoever's been following the mitochondrial metabolic theory of cancer, it's just been this slow chip away over time at, an accumulation of a number of different cancers. And where now it's like you've got Tom Seyfried accumulating this giant list of cancers that I think you said most of the blood cancers and epithelial cancers and tissue cancers all have some form of mitochondrial dysfunction, measured mitochondrial dysfunction, and
it's been identified.
It's been
But
identified.
probably it's varying degrees. I'm sure you can find some that looks okay.
These genetic cancers, these genetic based cancers or hereditary cancer syndromes that you've identified now, for a lot of people, this is it's promising because it doesn't necessarily mean that everything is out of their control, right? If it's metabolic, like you're saying, I forget the analogy you just used about fighting.
That impaired oxphos or impaired respiration, that may not be sufficient to cause cancer. But if it's necessary, if it has to be there,
right?
And from a prevention perspective, if you can maintain like the care and feeding of your mitochondria,
that's right.
Like that's critical. Like it is. It's like, to me, and I'm stealing it probably from somebody. Prevention is the cure, and that's why I think, like, I'm not kidding. The American Cancer Society or whoever it is, the most needle moving they can do is probably get people off smoking. Like, how any way they can get those rates down. That's probably how they make like the most impact. It's not through like trying to manipulate how well they're doing with treatment and screening. Just one quickie, because this is the thing this is the thing about reprogramming energy metabolism, and it's not like completely extensive evidence, but I think it's pretty compelling, is that I tried to look for, especially in these cases, they have all these like metabolomic studies, and they'll look at people. I thought they would look at this, but there's at least a few studies that did this. You have people who are BRCA1 carriers who don't have cancer yet, or just won't ever get a cancer, but they are BRCA1 positive for the mutation. Then you compare them to like all matched whatever cohorts of normal people. And then you also have, ideally, you would have BRCA1 positive who have cancer. So you have BRCA1 positive carriers don't have cancer. BRCA1 carriers do have cancer, and then you have a control group. Mm-hmm. And so when I looked for that, I found six analyses, or at least in 6 different of these hereditary cancer syndromes. So the way that they say it is like asymptomatic, that they actually looked at people who they've got Li-Fraumeni syndrome, but they don't have cancer. They have the P53 mutation. And actually, in this case, that's not one of the examples. So those mutation pairs, so the metabolic signatures in those people that don't have cancer had impaired, like effectively their metabolomics or biomarkers or blood, plasma, skin, blood, all that stuff. It suggested impaired oxphos, and it was moving towards the quote-unquote Warburg effect, SLP dependency compared to non-carrier controls. And so, like, these are the studies, and what these studies suggest is, how can your oncogenic mutations, including the ones in these hereditary cancer syndromes, if you're screening for them, it doesn't look like that's happening. It looks like the metabolism, the impaired metabolism, however you want to call it, impaired respiration, and potentially some of the compensatory fermentation that is preceding the mutations. Or, in this case, the mutations are already visible because we're not waiting for a somatic mutation. We already have existing germline mutations. And so the argument would be, you need to knock out the second allele. You need to have cancer before you reprogram energy metabolism. And the only studies I could find that actually looked at asymptomatic non-cancer carriers of these syndromes, they all exhibited effectively what looked like, they say, Warburg-like metabolism, I think a bioenergetic shift towards aerobic glycolysis. The Bononi paper, that one was like probably the most clear. It was like, these cells are quote unquote normal with they were heterozygous for DAP1. And they were showing like, and the ones that actually showed the cancer carriers, you could see that they were like, it was by degree. So you had controls relatively normal. You had carriers without cancer. Clearly there was a signature. that it was a combination of impaired respiration, compensatory fermentation. You had the cancer carriers that the degree was higher, and you think, okay,
yeah. Wow. You've got this striation with this clear, initiator in the metabolism.
Basically, yeah, you've got however they want to word it. You've got reprogrammed cellular metabolism, but you don't have cancer yet.
Right.
So that would similar to like the cancers without mutations. You would say, even like they have the mutation that may be conferring this, but they don't have the cancer.
Very
that shouldn't
interesting.
happen.
Yeah. Very interesting. Very interesting. Well, Bob, this paper is incredible. It's super, super important. It's for tons of different implications. We are super proud of you to have you as part of the BSI and Medfix team and family to say that one of our own published this paper, and I mean it's just fresh off the press, so it's only had a month's worth of impact, but I think that this is going to have a lot of impact for many years to come. And earlier you were saying the National Cancer Institute. I think you brought up the website. Cancer is a genetic disease. I think Greg would agree with us. I don't think there's a hope that they're going to change their view necessarily because of all of this evidence that starts to come out over time, for cancer being a mitochondrial metabolic origin. Mm-hmm. Because as Greg puts it, once you admit that that's the case, you're about 30 seconds away from saying that sugar causes cancer, right? Which starts to become a huge problem for a lot of different people. But by following the evidence and looking at this stuff, then it at least gives us a little bit of hope that maybe, simple environmental, lifestyle modifiable factors like diet, quitting smoking, etc., can have huge positive impacts for cancer outcomes. These are the controllables, right? Control the controllables. Yeah. And
I just think knowing nothing, I guess this is how I look at like the somatic mutation theory and how it's evolved, ironically, over time, is that like I have a heuristic now, which is, how does it work? The more you research something, right? And the more complex and multifactorial it becomes, like there's going to be at the beginning some level of that. But if you've been studying this disease for basically a century, and it just keeps becoming more and more complex and multifactorial, that can be used as an index for your understanding of the etiology of the disease. Meaning, like it's the more complex and multifactorial it becomes, the further away you are from the answer. And it's almost like the only way I can see these things is it's almost like if you are chasing an effect, this is what it would look like. Gary actually made that point, I think, in like Good Calories, Bad Calories. It's like if you hit somebody in the head with a hammer and you look at like all the cascades and everything else like that, like if you don't figure out that it's the hammer, is like you will spend a career looking at inflammatory cytokines, myokines, the immune response, immune modulation, epigenetic reprogramming, blah, blah, blah. And you're like, what about the fucking hammer?
Yeah.
That's what I think. Like this stuff. Like if you know nothing, that's why I think a lot of people who know nothing about this are at an advantage because it actually takes work, I think, to let some of that logic seep in about how to reconcile some of those challenges, particularly the nuclear transfer experiments. But yeah. Anyway.
So next time, Bob, we're gonna next time. So end of the month, we'll come back. It's not going to be live. This was a teaser for everyone who isn't part of the Broken Science Initiative Medical Society.
We're recording this?
We are recording this.
Okay.
Yeah, it's recording. So this was a live one, but next month we'll probably do another one, maybe closer to the end of the year. I figure a couple live ones a year is probably good. And but the next few months we're going to be only accessible for Broken Science Initiative Medical Society members and Medfix affiliates. And so if you're not a Medfix affiliate, become one. And if you don't own a gym or have a training business, then you can become a medical society member on our website, brokenscience.org. /medical will take you to the medical society page, and memberships are cheap. They start at less than $2 a day. So, but you're going to get access to a whole host of perks and education, but at the same time, you're going to be able to access our journal clubs that we do once a month, usually towards the end of the month. And so next month, Bob, we're going to continue on this thread, but we're going to analyze the challenges, the metabolic challenge of high-performing athletes, and how some of the metabolism, how the body uses metabolism in different ways during performance of athletics, and then sort of connect and contrast that to the way the metabolics of the cancer cell work, and start to see some similarities. And am I right on that?
I think so. Yeah, I think in part. Yeah.
Excellent.
I find it it's fascinating. The acute response, sort of what's going on when they look under the hood, if they can. It's difficult, but when they see whether it's exercise, especially when you go to high intensity, it's like you see this. It's almost like you could say like it's a hallmarks of exercise. And they literally, you look at MIC and HIF, BRCA1, P53, all these like skeletal muscle, like what they're doing is that all these, you could call them all, like many of them at least, important for like exercise metabolism.
Wow, very cool.
If that's the case, I think it supports, obviously, if you think this is like a mitochondrial problem, but in many ways, it's also a mitochondrial opportunity. Is that you can, a lot of the data seems to show acutely doing those things can like attenuate these things from a chronic perspective, but can also lay them up short term, which is not necessarily a bad thing. It actually means if you go super high intensity, you're actually better at burning the high octane stuff. I don't know if that's the right term for it, but
right. Amazing, guys. And if you're looking for, if any of you guys are listening and you're looking for some of the basics to be able to understand some of the stuff Bob is talking about a little bit easier, you can come to the Foundations course, the Medfix Foundations course. We give a basic overview of cellular metabolism, biochemistry that just sort of lays the foundation for being able to understand a lot of this, the deeper metabolism research and stuff like Bob's going through with the cancer research currently. So we've got if you're in Europe, we're going to Vienna in March. And if you're in the USA, we've got, or North America, if you're in Canada, you can still go over the border if you're in Canada. We're doing our next one after that in April in New York City. So hopefully see you guys there. And if not, we'll see you at the next Journal Club at the end of February. Thank you very much. Thanks, Bob. That was a pleasure.
Thanks, Pete. Thanks, gang.